So, good morning, everyone. It is indeed a great pleasure to welcome you all to this conference on Frontiers in Physics of Soft and Biological Matter. So, this conference is being held as part of the Platinum Jubilee celebrations of the Raman Research Institute. This institute was founded by Chandrasekhara Venkata Raman 75 years ago in 1948. Raman did his seminal work on what is now called as Raman scattering in the 1920s when he was at the Indian Council for the Conservation of Sciences, Indian Association for the Conservation of Sciences in Calcutta in the 1920s. And in fact, it was precisely 100 years back that the first observation of Raman scattering was recorded. In fact, one of Raman's students, K.R. Ramanathan, was studying scattering from water and alcohol. So they were basically using a very simple setup, a beam of sunlight passing through a liquid and looking at scattering with two complementary filters. And then they found that the scattered radiation had a component which they could not explain. It had a different wavelength from the incident wavelength. And this they initially attributed to weak fluorescence arising from impurities in the liquid. But Raman had an inkling that was something more than that. And he was thinking in terms of some analogy with Compton scattering, which is inelastic scattering of X-rays by electrons. But then it took them another 5 years to really figure out what was going on. And the discovery was announced in 1928. So soon after that, he got the Nobel Prize in 1930. And in 1933, Raman moved to Bangalore, to the Indian Institute of Science down the road. And he spent about 15 years over there. After his retirement in 1948, he moved to the Raman Research Institute. So Raman had planned in such a way that the day he retired from IISc, he could come over here and continue his work without a break in his research. Of course, the early days of the Raman Institute, there are very many interesting stories about those, which I'm sure which you'll hear from Professor Madhusudana tomorrow. The first 2 years, they didn't have any electricity over here. So all the experiments were done with sunlight. So they used to have somebody standing out in the sun with a mirror, trying to track the sun and having a beam focused onto the aperture in the lab over here. In spite of all that, they did very excellent work over those years. And Raman's main interest was in scattering of light from crystals, colors of gemstones and beetles and things like that, and also physiology of vision. Of course, Raman wanted complete independence for his work, and he didn't want to take any funds from the government. So he ran it with his own resources and also from donations from wealthy patrons. The land was given to him by then the Maharaja of Mysore, so he had the land already, and he constructed the infrastructure and carried on his research. He had a small set of students working with him at that time. Of course, Raman knew that after his time, things were going to change. He had sort of big plans for the institute. He wanted it to become a center where people would pursue different branches of science. And he knew that will not be possible without support from the government. But he was very particular that the institute maintained its autonomy as far as the research that was being conducted in the institute. Of course, after Raman's demise in 1970, in 1972, the institute became an autonomous institute under the Department of Science and Technology, Government of India. And the two groups were formed right at the beginning. One was the Astronomy and Astrophysics group, and the other one was the Liquid Crystal group by S. Chandrasekhar, which over the years has sort of diversified into different branches of soft matter and also into biological physics. So as we sort of celebrate this milestone, this conference is a good opportunity to look around and see the recent developments in the field and also look at important open problems that can be pursued in the future. Pramod and Sayantan have put together an interesting program for the next few days, with of course help from the rest of the group. And I'm extremely thankful to all the speakers who have come from far and near, who have accepted our invitation to participate in this event. Thank you all very much. And on behalf of the Institute, I once again extend a warm welcome to all of you and hope you will have a very pleasant and interesting week ahead. Thank you.
Thanks, Raghu, for the wonderful introduction. So now I'd like to invite Professor Prerna Sharma from IISc to chair the first session. Yeah,
welcome to the first session. Our first talk is by Professor Julia Yeomans. She'll be talking about modeling collective cell dynamics using active matter physics. So, Julia, please.
Okay, thank you.
Students and postdocs and everyone in the audience is welcome to ask questions in between. We have plenty of time in these talks.
Hello everyone, good morning. First of all, to say it's a real pleasure to be here. Thank you very much for the invitation. It's always wonderful to come to India and to be able to visit your beautiful campuses, and this one is really very stunning. I was walking around this morning and seeing all the beautiful gardens and trees. Please ask questions during the talk. I mean, going on for nearly an hour is going to be too much for me, so it'd be really lovely to have some feedback, especially from students, and possibly not too much from Shriram. Okay, so apologies for the advert, but our administration says I have to advertise this widely, so I've done it now, and I'll shut up for the rest of the week. So, I think we all know what active matter is. Things which take energy from the surroundings and are continually using it to do work. So active matter is out of equilibrium, and it's become a great excuse for doing non-equilibrium statistical physics. Living matter is out of equilibrium. That's what we're doing all the time. We're using energy to do things, and even on tiny length scales, the same is true. And so this has become a meeting point. Active matter has become a meeting point of physics and biology. And I think there are lots of exciting things going on at the moment, trying to understand how these two approaches to describing what happens in living systems are going to help or not help each other. So I'm going to talk about collective behavior of active systems. If you have a load of microswimmers and you put them together, what you end up is this swirly sort of pattern, this swirly sort of flow pattern, which has become known as active turbulence. If you, it's going to work, take microtubules driven by motor proteins, same sort of thing. You get a pattern where everything looks turbulent. Can't be inertial turbulence because we're at tiny length scales, but the same sort of things going on. And again, if you look at cells, these are epithelial cells taken out of the body and put on a Petri dish. And in some regimes, what happens before they get too dense and jam, again you see an active turbulent-like state in this system. So what I'm going to talk about is trying to worry about whether these are all the same, and in particular why the active turbulence in these cell layers, in cells which are joined to each other, are a little bit different from microswimmers. Is it the same? Can we use the same sort of theories to describe active turbulence in the cell layers as we can in the microswimmers? So the first part of the talk is sort of background, because I know that I have graduate students here who probably haven't seen this, and people from slightly different fields. I'll talk about a summary of active pneumatics, where this active turbulence comes from, and the fact that you get active topological defects in these systems. And then I'll ask the question, can cell layers be described as active pneumatics? First, by looking at the sort of motion you get in these cell layers, and then talking about very recent, as yet unpublished work about some experiments that come from Jacob Nottbaum's group. So, pneumatic. I think in this place everybody knows what a pneumatic is. Long, thin molecules. which have up-down symmetry but a preferred direction, and they can order into a nematic phase, where on average they have long-range orientational order, but no long-range positional order. And a very useful order parameter for describing that phase is a tensor, the Q tensor, and the largest eigenvalue of the Q tensor tells me how ordered these things are. These are perfectly ordered, but sometimes they're going to move around a bit. And the largest eigenvector tells me the direction of the ordering. So this Q tensor is what I'm going to use to describe the ordering in my active nematics. Now, in a passive nematic, one which doesn't have any activity, just a bog standard liquid crystal, if you have a localized distortion like this, which is going to happen due to thermal fluctuations, that's fine. It's not a big deal because the elasticity in these things is going to quickly take that distortion and push it back to the ordered state. But you can have more interesting distortions, these topological defects. And topological defects are places where the ordering is such that the directive field is confused all the way out to infinity. So in fact, a defect like that has an infinite energy if it's just sitting there on its own. And you can't unknot these things. You can't put them back to correct ordering without making changes all the way out to infinity. In 2 dimensions, these topological defects, the most common ones are these +1/2 defects, which look like a comet, and a -1/2 defect, which has these 3-fold symmetry. And what happens in a passive system is they behave like charges, so the plus and the minus defects slowly move towards each other and then will annihilate, unless they get stuck on a sort of dirt or something in the system. And really you can do without these, because if these appear on your laptop screen, which of course is a nematic, what it's going to do is completely muck up the screen. So this is a summary of how I write down the continuum equations for one of these passive nematics. Okay? I just put the sort of the main details here, the main ideas here rather than the details. So we've got a complex fluid. So first of all, we need an equation for the order parameter Q. Q is advectored by flows, as usual in these continuum equations. So, I mean, we're sort of doing things on a Navier-Stokes level here, but we've got this Q as well because things are more complicated in a complex fluid. This thing here couples nematic order with any shear flows, because if I have a nematogen like this and I have some sort of shear, what's going to happen is any sort of shear flow is going to turn it, and so you need to determine the equations to sort that out. And then you have a phenomenological term which says that I relax towards the minimum of the free energy, because if I turn off the flows, I want to end up described by the free energy of a system. And the free energy is the bog standard Landau-de Gennes free energy, which is an expansion in terms of the order parameter Q, plus a term which says I've got elasticity. These things don't want to be exactly lined up with each other. So then I need an equation for the velocity field. And the way I've got it written here, it looks exactly like Navier-Stokes. Usual advection term, which is very unimportant because things are rather small. And then I have the gradient of the stress tensor. The stress tensor has the usual terms that you have in the Navier-Stokes, which goes as the usual viscous terms, which goes as gradients of the velocity. And then another horrible term, which talks about the backflow, which physically says that if you have a liquid crystal and it's turning, it's going to move the fluid, and so it's going to induce flows in the system. So how do we take these and make them so that they're active instead of passive? How do we make a liquid crystal into an active liquid crystal? And this was one of Sriram's great contributions. I can be nice to him as well. Okay, and what he said was that what you have to do is you have to change the stress tensor. The physics that we're putting in here is the physics of these active particles. For example, if I have loads of swimmers swimming about, what they're doing is they're creating flow fields, which are dipolar flow fields, and that's giving you an extra stress. And it's not entirely obvious, so you're going to have to accept it at this stage, because else we'll never get anywhere, right? So this is the term you put in. This is the active stress. It goes in here. And the thing to notice is that it comes under a derivative. It's equal to zeta q, where zeta is the strength of the activity, how many swimmers you've got swimming around. This q is the order parameter. That comes in as q because of the shape of the flow fields in this system. And it goes under this derivative. Yeah?
So in this relaxation part, the Hij,
Yeah?
it doesn't contain the spatial derivatives of the director field. So is that not important? If you go back again.
There you go. That's the spatial derivatives of the directive
So
field.
so you are using the k as one constant approximation or something?
Yes. Yes. And in fact, if you have more than one constant, it doesn't really make much difference. If
Okay.
you have more than one constant
Okay.
there. Yeah. No, you're quite right. You absolutely need that term in to make it a proper liquid crystal. Sorry, other? Yeah? Okay, where was I? Well, where was I is the punchline. This comes in the stress. It comes under a derivative. So gradients in the magnitude or the direction of the order parameter give you a flow. And if you remember that, you can explain pretty much everything about active pneumatics. Now, I'm going to introduce now two words which we're going to use a lot during this talk. The first one is the idea of an extensile active pneumatic. And in terms of these swimmers, what it means is that the little swimmers push fluid out from their tails and pull it in from the sides. And in terms of the equations, it means that this zeta is positive. I'm also going to talk about contractile active pneumatics, which just means that the flows are changed in direction. And then this zeta negative is a contractile active pneumatic. More of that later. So we've got this extra term. What's it going to do for us? Well, a lot. In a passive pneumatic, a little localized distortion like that is fine. Nothing happens. But it's a change in the order parameter. It's gradients in the order parameter. That induces stresses and flows. So what flows does it induce? The active flows are in this direction. And so what they're going to do to this little defect is it's going to push it, and it's going to make it larger. And so the pneumatic order is unstable because the minute you have a small fluctuation, gradient in the order parameter, you get flows, which makes that fluctuation bigger. In maths language, these equations are unstable through a linear stability analysis. The other thing it does, and we'll see more of this later, is that the topological defects, as well as annihilating with each other, are also created in pairs and can move around. Let's see that happening. Okay? This is linear stability analysis, but then that can't tell you where we cross over to. We have to solve those equations numerically to see the active turbulence. So let's solve the equations numerically. This is a vorticity plot of what you get. The red bits are strong anti-clockwise vorticity, and the blue bits are strong clockwise vorticity. And so what you end up with is loads of flows which are swirling all over the place. And that's this active turbulence. And if I take this and I sort of zoom in to see what's going on, the black lines here are flow lines. I zoom in, and what we're seeing here is a jet, a strong flow jet. And these little short lines are this director field, And you can see that the director field is very bent here. I've got a big gradient in the director field, and that's what's causing the flows. You get this instability, gradients in the director field. This sets up this state with high vorticity and lots of jets of flows. What about the topological defects? Okay, so this is a picture, again, from a computer simulation where the red dots are +1/2 defects and the blue dots are -1/2 defects. And they're moving around just like they're meant to. And if I choose the right pair, they're busy annihilating each other in pairs. But the new thing which comes in here is that you get them popping out. You get defects created in pairs as well as annihilated in pairs. You're allowed to do that because it's an active system. You've got activity, you're pumping in energy. This energy allows you to overcome the elastic energy, which wants to line everything up, and create the topological defects. If two of them are just created and sit there, that would be useless because they'd annihilate again straight away. But what happens is that these things are self-motile. The +1/2s move away from the -1/2s. You can explain this because here we've got a defect, and there's pretty obviously gradients in the order parameter at a defect. Gradients in the order parameter give you stresses, give you flows, and so this thing can move. And it moves towards its head in an extensile system and towards its tail in a contractile system. So how these defects move gave us a way of understanding whether a system is extensile or contractile. These guys create flows as well, but because of the symmetry, those flows balance. Here's a picture of the flow field. The +1/2 has a flow field which has two vortices around it, and the -1/2 has a flow field which has this six-fold symmetry. So it's creating flows, but just sitting there. So a combination of the flow fields created by the defects plus the flow fields created by any sort of change in the order parameter, any sort of change, is going to give you this active turbulence. So that's active pneumatics. Gradients in the order parameter is what's causing the flows, and you get this active turbulence, which has this new thing about a gas of topological defects. Yeah.
So I was wondering that you are calling it turbulence. Does it have any energy cascade like the passive one, or like?
No. To a good approximation, no. I mean, you can see scaling laws, but it's very much not an energy cascade, because the energy here has been pumped in as this tiny length scale. So turbulence, if you're a physicist, was a lousy word, in fact, because it's caused confusion. It's more turbulence, as in someone on the street would call turbulence. Yeah.
Okay. Because of this existence of vortices and the randomness of the flow, it is called turbulence, isn't it? The active.
So I didn't get the question there.
So what I was thinking that because you have vortices, that is they are also in turbulent flows, and the randomness of the flow, that's why like it is called active turbulence.
It's really the randomness of the flow.
Okay.
Yeah, yeah.
Okay.
Active chaos might have been better, but yes, the vortexes have a particular length scale which is set by the elasticity and the activity.
Okay.
Yeah. Right, now it's
Thanks.
a really good time for questions, actually, yeah. Other people have questions. Steve?
You were suggesting that these ideas would also apply to cells or maybe to colloids. Where does the pneumatic correspondence come from?
Good question, and that's coming in a minute. I mean, that's exactly why you think maybe they don't apply to cells, because they're not obviously pneumatic. Yeah. Yeah.
So you mentioned that when the opposite pairs were created, they're infected away from each other.
Yeah.
But then you also showed that
No, they sort of vect themselves away from each other. Yeah.
they vect themselves away from each other.
Yeah.
But ultimately, they annihilate. So are they coming together in a different orientation when they annihilate, or how do they ever manage to annihilate instead of just swimming away from each
Because they're annihilating with a different one.
other with a different one?
Yeah, so they're all moving around. You've got this gas of defects, and just by chance
By
they
chance.
hit someone. Yeah. Mm-hmm. Yeah, Joseph? No, I didn't say it doesn't
Okay,
apply
so
to
I
cells.
just got
said,
it wrong.
I said you have to worry about whether it applies to cells. See the rest of the talk.
So if I look at the previous slide, if I look at the flow field around the defects, it looks like it's neither contractile nor extensile. So what kind of systems are these?
Because this is the flow field around a topological defect, and the contractile and extensile refer to the flow field around a single particle. So that's why you can't see it exactly from here. It's the way this thing moves, like that or like that, that tells you which it is.
So you have these defects, red and blue, which are moving.
Yeah.
And, so is there a simpler effective dynamics for the defects if I don't focus on the underlying flow?
We've tried, lots of other people have tried.
Okay.
I don't think we're there yet with that one.
Okay. So
Yeah.
it's
because it's a non-equilibrium system, that's why it makes it very hard.
but like some active kind of dynamics.
I don't think that anyone has written down something totally convincing
Okay.
for how these things, as a gas of particles. Sorry, you'll have to shout.
So I was asking, the particles themselves are not moving. They're just shaking the fluid. The defects are the ones that are doing the moving.
The underlying swimmers are the things that are moving. Okay?
Okay.
These are also moving. These, thinking of a defect as a particle. But otherwise, we're doing a continuum model here, so it's sort of all the underlying swimmers are making the flow field. Just like in Navier-Stokes, all the underlying molecules are moving.
Thanks.
In terms of fields. Great. Okay. Thank you. It's great to have questions. So now let's ask this question. All right, so let's think a bit about what we're talking about to start with. We're talking about epithelial cells, ones like this. And a single cell can, if you're a physicist, be thought of as just a sort of nucleus with these green actin filaments, and it moves because the actin filaments are pushing it forwards. It grabs onto the surface through focal adhesions, and it pulls itself forward. So a simple model of that, right, I apologize at this point for any biologists in the audience, is that you have polar driving, just a force pulling it forwards, and then so it doesn't leave its tail behind, self-contractile activity when it pulls in its tail. And that's a pretty good model because these things do a persistent random walk, and if you don't really care about the biological underpinnings, that describes how they move. Now let's put them together into a cell layer. This cell layer has the cells attached to each other through adherence junctions, so the cell boundaries pull on each other. These are epithelial cells, and they're really quite a common sort of cell in the body. They tend to line organs, and so they do tend to make two-dimensional layers, and we always look down on them when they're stuck on a Petri dish, and so I'm afraid we tend to think of them in two-dimensional, although that's really an approximation that needs to be worried about. So these things move. It looks chaotic, but why on earth are these active pneumatics? Because on average, they are circular. So let me summarize the ways in which these cells move collectively. First of all, they can jam like this. If they keep dividing, they get dense, and then they jam. They can also flock. They can suddenly decide to all take off in the same direction. All right? This is the inside of the egg chamber of Drosophila, and the cells just decide to move round like this for reasons no one, I think, understands. This is another picture of those cells moving round and round. Yes, Joseph?
Model A is already confluent before. In the pictures where you still, so the cells moving, they're already at volume fraction 1. So in that sense,
But they're still being pushed, they're becoming smaller. Remember, we've got the third dimension here. So cell division, if cells divide, what happens is that these things jam.
yeah, but I think it's more shape-induced than actually density-induced.
Okay, well,
I doubt that it's density-induced.
okay. Okay, so we get this flocking. You can, yep,
So the video where you show the flocking behavior of these cells,
yeah.
are also the internal machineries kind of in sync to give you this flock state, or is just one cell pushing, giving rise to a collective growth? So what happens, is there coordination at the level of the cell, across cells?
Let me give the physics answer to this, that if you want to model that, the way you would do it is a VTREC model, which is that you have alignment of the cells with each other. Why cells actually flock in terms of the underlying biology is still something of a question. Yeah. Okay, and also these cells you see active turbulence. So what's the evidence for that? Well, first of all, if you look at the velocity field, the velocity field is very reminiscent of active turbulence. Here I've just shown pictures which compare the experiments with the simulations, but you can look at correlation functions and do better than that. And you see these jets, and you see the vortices. People have now found topological defect in all sorts of cells. This is a movie showing, whoops, the next one's a movie, showing plus and minus a half topological defects in a layer of human bronchial cells. Lots of them, okay? And they've now been seen in many, many different sorts of cell systems. This is our original paper where we found topological defects, much to our surprise, in MDCK layers. MDCK layers are very much these round, squishy cells that I've been showing you the movies of. So the first question is, quite rightly, as I was asked, why on earth are these things pneumatic? And the answer is that locally these things fluctuate, so they're extended. And then if they're extended, they're stuck to the neighbors, and so they tend to pull their neighbors. And so you get little patches of pneumatic order. And when you take the length of the cell, the long axis of the cell, and you just put a director, a line along the long axis of the cell, and then you plot them, this is the sort of picture you get. And you can see pneumatic order. It's not long-range pneumatic order, but there's definitely short-range pneumatic order in these systems. And you also find topological defects. This topological defect is a well-identified shape which persists over hours and which moves towards its head. Yeah?
So if these were two-dimensional objects that had a similar aspect ratio, would they also have pneumatic order, or is if there was no activity, like is the enough of
No. If there was no activity, it would be like a foam, and the ground state would be a load of hexagons.
but these you said are slightly polarized.
They're polarized because they're active, so somehow the activity is making them fluctuate. So they're polarized, but it's a fluctuating polarization. So on average, they would be round, but the fluctuations are slow.
If you had a thermal system that had the same distribution of shapes,
No,
it would not be
it would not, because thermal means Maxwell-Boltzmann, and these definitely are not Maxwell-Boltzmann.
Okay,
Yeah.
thank you very much.
Yeah. Okay, so this is a nice experiment, which I also, yeah, sorry, go on. No.
Yeah, so I'm just wondering if this in the ground state, is there any orientational order in the system or it is isotropic?
Ground state's a funny thing to say because this is an active system and it never makes it to a ground state, okay? But if it did, it would be this hexagon of foams because they're stuck to each other and they're sort of space filling.
Okay.
Yeah.
Okay.
Yeah. Just to prove, this is an experiment which I think is a nice bit of proof that these are actually active defects, okay? Number of defects in this layer as a function of time. So what the experimentalists, and I should say this is Thuan Ben So and Benoit Ladou's group is the experimentalist here, he added blebistatin, sleeping pill for cells, made them passive. The number of defects went down because they were annihilating with each other, washed out the blebistatin so they became active again, defects started being made, and the number of defects went up. Extensile. That's weird, isn't it? I said these things are extensile. I said these things went towards their head, so they're extensile. I also said that cells tend to be contractile. Everyone knows that cells are contractile. They don't sort of suddenly, as they're moving, a single cell doesn't elongate itself, right? So a single cell is definitely contractile. It sort of moves like this. So there's a big question here, which has been worrying us for ages. Are cells extensile or are they contractile? I'll park that and come back to it. But first, let me show you what happens when you take this stuff and you put it in confinement. So you confine an active system. It is known that when you confine an active system, you destroy the active turbulence, basically because the vortices can't fit anymore. And what you get instead is often just one vortex and regular flows. And the same thing happens with cells. When they're confined, you start getting this regular flow. You see that in many different active systems. And then there were some beautiful experiments from the Paris group. They looked at confinement in a channel. So these are cells confined in a channel. They're active, so what happens is they take off and they start flowing. You get a shear flow. They're an active system, they've got energy, they've got to do something with it, so they have a shear flow. And what's particularly nice about this is that you can do the theory because of the simple geometry, and there's an almost exact match. The almost is that these things are slightly chiral, but it's an almost exact match between the theory and the experiments. And happens in macroscopic systems as well. Macroscopic active systems will rotate.
Julia, did they ever repeat this experiment?
Yeah.
The other way.
Oh, I don't know actually. The videos like this all over the web, so it's a good thing to see. I'm not going to talk about this bit. Yeah. So we try to answer the... yes, okay. Yep, now it's a good time for questions. Yeah, yeah, please do.
In your equations, you had this Frank elastic free energy kind of term,
yeah,
where this play, bend, or any of these distortions cause energy. In the case of cells, is the physics similar, or because these are now the shape itself is not anisotropic?
That's right. I mean, the answer is that's a complicated question. I'm not sure I know the right answer. The cells definitely hang on to each other. If one cell is elongated this way, the neighbors tend to be, because it looks pneumatic. It's not long-range pneumatic order, so actually these topological defects are not quite topological. But exactly what the biological underpinning for that is still a big question. Yeah, so it's a good question. I'm not sure I know exactly the answer, but certainly it looks like an active pneumatic. Mm.
So when you showed this plus half defects in your cell monolayer, does it still follow the same sort of creation dynamics that you would see in a pneumatic, or is it something very different?
It looks the same. We at this stage in the experiment, I mean, I'll show you something a bit different later, but as far as we can see, the topological defects behave in the same way. Yeah?
So the flow, in the channel flow,
Yeah.
you showed the flow field. Looked to me that it is far from simple shear flow, because it looks like there is very strong banding, and also you said that the theory can describe it. So the shear bands, like, does it come from the microscopic theory?
Yeah, the theory is just the equations of motion that I wrote down there, which have been solved exactly in one dimension by Raphael Voituriez and Jack. Jack, were you on that paper? Probably? Yes. Yes.
Actually, we predicted that behavior more than, we predicted that behavior more than 10 years before the experiment was done.
Mm.
And this is, I would say basically Shriram active nematic theory. And you start from something which is well aligned over the whole sample. And if you get to larger width, then you get spontaneous transition to a simple shear close to the transition. And if you go away from the transition, then you can see other terms like the interaction with the substrate and stuff like that. But as long as you don't get to the turbulent regime, you can understand basically everything. And
yeah, I think that's why it was such a beautiful experiment, that it really nailed the theory, yeah.
But just following up on that, are there shear bands in these flows?
No.
There are no shear bands.
I mean, there are in some, not in this one here, no. Okay. So then, I got this question, are the cells in confluent cell layers extensile or contractile? So what I'm going to do now is talk a little bit about extensile and contractile. Just to remind us, extensile forces, the flow is outwards from the ends and inwards from the sides. Contractile ones, you just change the arrows and have it inwards from the ends and outwards from the sides. In the equations of motion, that zeta q term changes its sign. I want to think about that. For cells, it's a little bit different. For cells, these cells, it's a little bit different. And the reason it's a little bit different is that these are squishy cells. So if you pull them, you actually extend them, as well as just sort of pushing the surrounding fluids and making them swim. So what I'm going to call extensile is when the mismatch angle between the principal axis of contractile stress and the shape axis is small. So what I'm on about here is that this is a contractile cell. It's pushing in here and outwards there, okay? And what it's doing to this shape is, sorry, this one's, let's get this right, the contractile ones. This is the contractile axis. It's the one pushing in, and what it's doing is it's squishing the cell, all right? And the angle between the axis of principal stress and the axis of the cell shape is just 0. And in general, I'm going to call a contractile cell where that angle is small. Extensile forces, on the other hand, elongate cells. Here, the axis of principal contractile stress, the direction with which you're squishing this cell is here. And it is perpendicular to the long axis of the cell. Okay? So what I'm worrying about here is the angle between the axis of principal contractile stress, the angle on which the cell has been squished, and the angle at which the shape changes. Okay?
Julia, quick question.
Yeah.
Can I then model this as an extensile force dipole, and that one does nothing? Because can I imagine that the shape acts like a force dipole pushing on the other cells?
What I want to do is decouple the force dipole and the shape. That's really what I'm trying to do here.
Okay.
That's what I want to do. I want to have two director fields, one for the shape and one for the stress. And the way to think about it, I think, is that extensile forces elongate cells, and this angle is small. Sorry, contractile forces, I put that there and it's meant to go down here. That was bad. Contractile forces restore the cells to circular, and the mismatch angle is small. Extensile forces elongate shells, and this mismatch angle is sort of large, near 90 degrees. All right. So we think cells are contractile. So the contractile ones were the ones which restored the cells to circular. And you've all been telling me that if they're circular, they can't be pneumatics, and you can't have active turbulence, which is dead true, okay? So if these are really contractile forces, you're in a mess. So a lot of people have spent a long time trying to understand how you might have extensile forces in these cell layers, and there have been lots of good ideas. Certainly cell division acts as an extensile force. There's a beautiful old paper now which showed that. If you have lots of dividing cells, because they're pushing outwards, they act as an extensile force. Some nice work by Farsenbaffer and Killeen in Imperial have shown that if you have fluctuating polar forces, because the fluctuating polar forces are going like this, and then like that, and then like that, and then like that, when you average them, it's like an extensile pneumatic force. And then we showed that if you have interactions between cells rather than in a cell, actually what happens at the boundaries means that even contractile interactions will extend cells. But that's a different story, one that I'm not going to talk about today. Sorry, can we leave questions on that bit till the end, because it's slightly tricky, and now you can forget about that bit if you like. What I want to show, and I'm going to show you experiments and theory to show this, is that it's wrong to call a cell extensile and contractile when it's these squishy cells, because the mismatch angle between the stress direction and the shape direction can change. So that's going to be the conclusion. Let's have a look at the evidence for that. This is work we've done with experimentalist Jacob Notbaum and Molly and Jay working in his group. In Oxford, it's been fronted by Marana Nejad, who's going off to a postdoc at Harvard very soon, submitted her thesis yesterday, which is a pity because it's horrible to lose. And Liam and Guanming have also made significant contributions. Okay, so these are the experiments. Yeah, sorry.
Hello? Yeah. I thought that if you have a closed body, three-dimensional.
Sorry, I'm having trouble hearing, so can someone say? Yeah.
My question is, isn't that some theorem that, if I have a compact ball, yeah, and I put the tangent vector field around it, on this surface tangents I plot of this ball, on this ball, then you find that at any given point the principal radii of curvature are always orthogonal.
Sorry, I'm not getting that.
We'll maybe discuss it.
So I put a ball, yeah.
No, I have a two-dimensional surface.
Yeah.
And I just plot its, you look at the tangent vector field on it, unit tangent wheel.
Tangent vector field.
Okay.
Just obtained by, so the principal radii of curvature, I have always an axis, which is like that contrariety and contrary.
I think it might be that you're thinking about equilibrium systems.
No,
I mean,
this is a mathematical fact.
Mathematicals, okay. Perhaps you could explain to me afterwards?
Sorry,
I'm not.
I'll attempt to do that.
Yeah, thank you. where was I? Okay, so this is just one of these MDCK layers. And what the experimentalists did is they measured the nematic shape axis of the cell. And that's easy enough. You just look how long they are and which direction is the long axis. And they also measured the direction of principal contractile stress, i.e., we're assuming this is a dipole, and is it a dipole with force, major force in that direction or that direction? You can think of it as the direction in which the stress fibers act for now. And they did that with traction force microscopy. So we know the forces on these cells, and we know the shape axis of these cells. And what they found is that there are regions, this is the layer, this is a part taken out from the center of the layer, red regions where this angle between stress and shape is small, i.e. the red regions are contractile, and the blue regions are extensile. You have large mismatch angles. The cell is not pushing out along its long axis, it's pushing out along its short shape axis. These regions change in time. This is a time evolution. And although I've made them contractile and extensile, the angle, the misalignment angle, is actually not just 0 or 90. It's a range between 0 or 90, and I put the cutoff at 45 so we can see what we're doing. Let's look at the movie of that. You can see that these regions are evolving, the extensile regions are evolving in time, and then relaxing back to be like the contractile background. This is the same, whoops, what's it done?
Just
Should I start it again?
No, duplicate screen, we can make it. Just
How can I do it?
Just a second.
You do up there, slide. Oh, sorry.
From you, right?
Yeah. Yeah, okay. So what I'm showing here is the distribution of the mismatch angle. This is what I called contractile. This is what I called extensile. So these are just the same story, but with sort of slightly more data. And we did this for 11 different layers. And always this was the area fraction of the contractile ones, and this is the area fraction of the extensile ones. So cells are changing from contractile, which you would expect, to extensile and back again. Another way of saying the same thing, not using contractile extensile language, is that the direction in which they're exerting forces and the direction their shape is extended in are usually the same as each other, contractile force along long axis, but can also be perpendicular to each other. And the reason is that, I mean, you can see if a topological defect moves through this system, this is a contractile defect, it's moving in this direction. These are the shape axes where the cell is extended. It's moving in that direction, okay? And so if you're in front of it, the shape is in this direction. And if you're behind it, the shape axis is like that. So the flows which are moving this defect are also changing the shape axis. And they're also going to be changing the stress axis, but there's no reason why they're changing it in the same way. So how would we model that? Well, these are the equations we've had all along. This Q-tensor told me about the order. I'm now saying I ought to have two director fields to model this system. I need a shape director field and a stress director field. So I need a Q-tensor for each of them. So rather than having a single equation here, okay, I end up with two equations which are the same as each other, except one's for stress and one's for shape. What's driving this thing? Well, it's the stress fibers which are driving it, and so I put the Qij stress here. The shape has no driving, it's just driven by the flows created by the stress. And there's one more thing we'd put in to model it, which is that we change the elastic free energy, so we have a different K for stress and shape, and that means that because you have different elastic constants, the shape and the stress are going to respond to the flows in different ways. And then we have one extra term that aligns stress to shape. Okay, that means that if it starts off as contractile and becomes extensile, then the stress relaxes back, so it's parallel to the shape. If you do that, you can get exactly the same thing happening. Top is the experiments, the bottom is the simulations. And you can see that, you know, at least average out, those things are pretty much exactly the same as each other. So we can also match things quantitatively, but that's slightly more iffy because there are a lot of parameters. But I think the fact that we get the same thing happening at least shows that in these models, in this extended version of the model, you can get this sort of behavior. We then worried about topological defects. In both the experiments and the simulations, we noticed that the defects tend to sit at these interfaces. They tend to sit at places where the mismatch angle is about 45 degrees. So we measured the number of defects at an interface. We had to define how wide an interface was, and the yellow things tell me the area fraction of the interface, and the green is the fraction of defects that sit at the interface. So again, in both the experiments and in the simulations, what you end up with is something which is doing the same thing. So what are these stress fibers doing? Okay, I believe in the simulations, we know what we're doing. I believe in the experiments to the extent that traction force microscopy, I'm never quite sure what it's measuring. But you know, what are these stress fibers? Why are these stress fibers pneumatic, and why do they align with each other? That I really don't understand properly at the moment. What we know is that if we measure the magnitude of the stress against this misalignment angle, if you have a nice contractile cell with the stress and shape aligned, you get a really good stress, but this goes down as you have this misalignment. So somehow it looks like when things are misaligned, the stress fibers aren't so efficient, and they're busy trying to reorganize themselves so they're in the same direction as the shape.
What does the magnitude of the stress mean for you? Like what part of the stress tensor?
It's the traction. Why do I think? I mean, it's
a tensor. So what am I using as a scalar?
You are using, it's a traceless tensor, the principal direction of contractile stress.
So the principal eigenvalue.
Yeah, the principal eigenvalue. That's right. Yeah.
The Qij shape and the Qij stress coupled to each other apart from via the flow?
They're coupled in two ways. One is via the flow because the stress makes flows and they both respond to that. The other one is this free energy term, which tends to take the stress and shape and relax them towards each other.
But there were cross-coupling terms? I missed that.
Sorry,
Were the cross-coupling terms Qij shape and Qij stress?
No, those are the only two. in the equations of motion, the only cross-coupling term is this one in the free energy. Yeah. Okay, so I think, you know, we're pretty much near the end now. Just the very last thing is that we then got told by the experimentalists we have to do it on a different cell line. These are long, thin cells, very different from MDCK, and they behave in a very different way. You get these misaligned areas, but they're very small, and they don't move. Okay, so these are typically misaligned areas. They're stuck there, and you hardly get any of them. The way we modeled this was we looked at the shape director, and we saw that it didn't change. In the experiments, the shape director, this is me pressing the wrong thing. Okay, okay. Anyway, remember this, the shape director doesn't change. So what we did in the simulations is we fixed a defect in the shape director, but we didn't allow it to change. Okay? And we saw what happened. And the answer is going to be, in a minute if we're lucky, that you get exactly the same story in the experiments. You get this little blob of misalignment. You get the shape director here, and you get the stress director a little bit away from it. The defect in the stress director organizes itself, so it's not in exactly the same plate. It's a little bit away. This is the simulations. Remember, the shape director is fixed. The stress director can vary. And what happens is you get this misalignment region, which doesn't change much in time, where the defect in the stress and the defect in the shape are not on top of each other.
Quick question.
So what is happening, one minute. I'll stop in a minute, then you can have it. So what is happening is that you're getting a competition here between the flows which are acting on the stress director and the fact that the stress director is trying to realign to the shape director. And the competition comes out like that. And in fact, exactly where you get this blob varies, depends on the initial condition. This is actually a metastable state. Exactly where that defect comes ends up being metastable, both in the experiments and in the simulations. So, yeah, I think this active pneumatic theory is giving us a lot to think about in terms of these confluent cell layers. But we think we need to decouple shape and stress. In the simulations, we can do that easily, and it's turning up all sorts of things which do look like the experiments. But I really don't understand. I mean, it's obvious when you look at shape what pneumatic ordering means, but there's a lot to be understood, I think, about what it means talking about pneumatic order in the stress. Thank you for listening, and very happy to have questions now.
Okay, so what is the difference between the MDCK cell lines and the new cell lines that you have, like in difference in terms of modeling? Because one seems to be an elongated cell, other is a circular cell.
The biggest difference, I mean, we do use different parameters, but the biggest difference is the way we put in the elastic constant K.
So they are anisotropic. No, you cannot make them one constant.
No. It's just, they're going to be stronger. It's going to be a stronger K if they're sort of, if they're long, thin things, which really do look like a pneumatic, okay? The cell-cell interactions are going to be different, and that means a different K. So in the model, what we do to go from one sort of behavior to the other is change K, but also actually fix the pneumatic director. It's frozen in, all right? And that's just because that's what happens in the experiments. Yeah.
Beautiful talk. Marvelous talk. So at first, one specific thing, Herb Levine's modeling shows that most cells of the cell lines basically always have a fraction of more epithelial-like cells and a fraction of more mesenchymal-like cells. Epithelial cells basically have more contractile ring, so basically they have an azeotropic contractile ring, while the mesenchymal cells have stress fiber-based contractility. So couldn't be your result simply that where you have the bluish area, you have some more epithelial cortical contractility, while you have in the other areas the stress fiber-based contractility. So is it?
It may well be related to that transition. It may be that what we call contractile and extensile are what other people are calling mesenchymal and the other one. But
I like contractile more.
But
sorry.
I am sure that it's more than just that. I mean, there are lots we have to understand. They change from one to the other as well, which is fine. And also, you have to be a bit careful because in the bioliterature, contractile means something different. Contractile means the forces that are pulling the cell inwards.
Yeah,
Big surface tension, which is different.
Which would be epithelial? Which would be, yeah. So,
Yeah,
okay.
but maybe, yeah.
Second question is a more fundamental. As a helpless experimentalist, there are basically currently for the same experiments, there are two worlds of theory. There is the active pneumatics, which is definitely true, and there's the whole shape-induced unjamming world, which is quantitatively also true. And you basically get pneumatic streaming for free because otherwise they couldn't move. Do they go together, or are they in? So currently there is large and cool stuff about active pneumatics. Then there's Lisa Manning, and somewhere I can't put it. I feel it has to be put together.
Yeah, we're working on it. We're working on it. The big difference is that the basic Lisa Manning story is not active. You can put in active polar forces, i.e. ones which push it like this, and you can in these continuum models as well, and then you get flocking. What people haven't done much of is putting dipolar, this sort of active pneumatic-like contractile extensile forces into the Manning-type models. We've started to do that. You see active turbulence, but there are all sorts of interesting questions because pneumatic interactions in a cell or pneumatic-like dipolar interactions between cells have different effects, and yeah.
Julia, am I allowed to ask a question? I kept almost completely quiet during.
As long as it's a friendly one. Yeah. Not that you would be anything else, yeah.
No, I have a couple of very different questions, actually. One is, let me ask the sort of short one first, which is that when you coupled the Q tensor, the stress Q tensor and the shape Q tensor, you did it only in a free energy. In principle, you could have done it directly in the equations of motion, in which case their coefficients could be violently, opposite. You could even have opposite signs, and you could get oscillations. If you couple them in a free energy, you can only either get relaxation or an instability.
I'm not so sure.
That is
no, I mean, because the zeta Q term is just in the stress.
No, I know. I'm saying no, but in the equations, the point is that when you introduce a coupling between Q shape and Q stress in the free energy functional, that limits the kinds of coupled dynamics they can have in the equations of motion of the two Q tensors, because that coupling is reciprocal.
Mm-hmm.
If you just wrote it down in the equations of motion and said, look, I'm not constrained by free energy, I can have, for example, opposite sign couplings of the two. You can have very non-reciprocal behaviors, including pursuit and rotation and so forth. I wonder if you've thought of that. Is there any evidence that the two alignments possibly even chase each other with a time lag? That's one question.
We have seen that in a 2Q model where we weren't thinking in terms of cells. Yes, you do see that.
Okay, my
Saraswat
other,
has been doing work on that.
I see, okay.
And we'll be
Okay,
pleased
maybe
to
I
tell
can
you about
talk,
it.
okay. So,
but here I'm not so sure about the physics of that. I mean,
Yeah,
what
I
I would do, the first thing I would do is change the alignment parameter in the two equations, and that would certainly give a different response to the flows and would be an
Sure.
alternative to changing K.
Yeah, okay, that's true.
Yeah.
Okay, my other question is completely different, which is that, these are experiments on cell layers on a substrate.
Yeah.
the forces aren't obliged to be divergences of a tensor because you're not constrained by momentum conservation. The flow fields around a given motile object aren't constrained to be like the flow fields in bulk, which are four-lobed. In fact, one of the pictures you showed early on really had a two-lobed flow field pattern around a plus half.
Yeah.
So do you need, I mean, so therefore, especially for these experiments, is this notion of an active stress proportional to the Q tensor rather than just a force monopole plus a momentum sink? Well, could that suffice? And is that what's at work, for instance, in the Manning way of modeling?
I mean, that's a really good question. I don't know the answer. The traction force microscopy fits reasonably well, thinking of it as local dipoles. I wouldn't believe it at all if our equations didn't give the same thing,
Right.
assuming that. But I think there's a really big question at the moment about, in these monolayers, whether you have dipoles. There's a thing called contact inhibition of locomotion,
Right.
which says that cells turn off their polar forces when they're stuck with each other. But then that nice work by Christina Marchetti and co and by Chufan Li said that actually what happens is that you do have polar forces, they just fluctuate, and so it comes out as dipolar. So,
I see.
so
Okay.
I think, I don't know yet, I'm not,
Thanks.
yeah.
So, bit biology-related question. So, for the LP9 cells, where you see the pinning of this defective region, right? Is there anything happening biologically which is different from the other regions? Is there a separate, special kind of biology happening there?
I would say, I mean, that's a really nice question. Maybe looking at one of these regions would be a way to see if there's something weird but biological going on. But we really don't understand the underlying biology. Apparently, the experiments are really hard looking at stress fibers. That's a pretty tough thing to do. Yeah. But I think that's a nice, you know, maybe focusing on those regions might help. Good idea.
Yeah. Okay. So when you describe, you know, extensile versus contractile, as Joseph said, there is also an isotropic piece, which can be entirely contractile, depend on also on system. But so you've just been speaking of the difference between the two directions.
We're incompressible.
And globally, it could still be contractile in all directions.
Yeah, no,
Right, that's a point. And,
at least in the model, we're incompressible, so we don't pick that up.
even if it's incompressible.
Really?
You know, well, it will not manifest itself, but it can still exist.
Yeah, I'm sure it can exist, yeah. in reality.
And the other point is that the fact that there is somehow some ambiguity when you define a nematic order parameter also exists in equilibrium system. You define the nematic order parameter as the anisotropic piece of the magnetic susceptibility or the TB, for instance. They need not to have the same eigendirections.
Okay, that's interesting.
And so that's, and actually your example is beautiful because there you can measure it. In a thermotropic system, it's very hard to measure it. And in fact, that doesn't change the long wavelength description, but it does change the so short range description. And what you do here is show that this short range is not so short range because you can see it. And that's the beauty of it, I would say. But I agree with Sriram that then, since it's out of equilibrium, it's not, and the dynamics might be very fancy, and you might see oscillation and stuff like that one day. I don't know, but it's nice. Yeah. Thank you.
Hi.
Sorry.
Yeah. You mentioned that in experiments, the shape tensor field remains more or less constant. Can you comment on how constant, like how fixed it is? Like, is it completely quenched in space, or is there some wiggle room where the
In the LP9s, it is fixed on the time scale of the experiments, which I think is several hours. Yeah.
Is this motion is always turbulent, or is there a driving parameter or something where it will be kind of steady? Or so I see all the pictures are kind of turbulent. So is there a regime in which it's not turbulent?
No. This is what Shriram showed, that it's unconditionally unstable in an infinite system. But in a finite system, then you do tame the turbulence.
Okay.
Yeah.
Just concerning the first dipoles, because I know it's a little bit a different breed, but experimentally, there are really two types of cells. There's the cells which basically form stress fibers and form force dipoles, which now I agree it's a bad language, but there are more mesenchymal, and there are the epithelial cells which have only cortical rings and have no stress fibers. So in that sense, I think there is a clear concerning Shriram, there's a clear divide in these cells. There are cells which basically which are able to be polar, and other cells which are not able to be polar.
Okay, well, I mean, I think that's a very interesting point. And I tended to think in terms of stress fibers when I was giving this talk, because it's much easier to visualize what I'm on about. But it's perfectly possible that these dipolar forces are coming from interactions between cells. The evidence against that is we do see a difference in the stress fibers, at least in... Actually, is that quite true? Well,
but you naturally...
Yeah, we do.
You picked cell lines, you picked exactly the cell lines you wanted for that.
Well, actually...
You didn't use a whole breast cancer cell panel where it switches.
I'd love you to do some experiments, please, with other things. I mean, we saw these experiments, and actually the first thing we saw was all the defects sort of sitting on these boundaries, and then we tried to explain them. We'd love some more experiments because there's so much I'm sure we've got wrong.
And 2D naturally has a great advantage because the nucleus doesn't play a role, because it sticks out. It actually gets much more complicated in 3D.
Yeah.
Okay, let's thank Julia for a wonderful talk and break for coffee. There's an announcement.
Just a quick announcement about drinking water. We have not supplied bottled water, but there is a reverse osmosis water filter right here at this corner. So please make use of it, and you have water bottles in your bag. So, yeah. And for students, the poster boards are all there. So anybody who wants to present poster, feel free to put it from day one, so that you can use it to interact with the speakers and so on. Yeah. And we will assemble by 11:40. Is it 20 minutes, no? Yeah.
Professor Eric Dufresne, tell us about putting the squeeze on phase separation. Over to you.
Okay, I'll get started. Yeah, thanks for coming. I am very happy to be here. This is my first trip to India. In fact, I only arrived this morning at 4 a.m. So I might be slightly less coherent than I usually am, but I'll give you my best shot. Today, the work that I'm going to tell you about is a project that spanned most of my time when I was a professor in Switzerland. It started by experiments with Rob Stile and Sai, was continued by Katie, and then the data you'll see in the second half of the talk was all driven by Carla, who's a postdoc who's looking for a job. She's amazing. So to start off, I'm really excited to tell you a local story here. And in many ways, the story starts in Bangalore, actually, and so it makes it especially nice for me to tell you the story. So this is a bird that I've only seen on the internet and in X-ray scattering and an electron microscope. I'm hoping to see it in real life sometime this week. If any of you are bird people, please come. Oh, you? Okay. No. I would love to see this bird, okay? So this is the Indian roller, and it has really spectacular colors in its plumage. And I'm not the only person who finds the colors of this bird to be really amazing. In fact, this guy, C.V. Raman, was also fascinated by the colors of the bird feathers in the Indian roller. And in fact, when he created, or I don't know this history, so someone can explain it to me, but I understand that he was instrumental in the founding of the Indian Academy of the Sciences. And one of the people that were founding the Indian Academy of the Sciences, and in Volume 1, Issue 1, Page 1, the very first, in Part A of the proceedings, was this article by Raman about the origin of colors in the plumage of birds. And so basically Raman what he did was he took a feather and he looked at it in a bunch of different ways, primarily through a light microscope, and also looked at how the colors of the bird feathers changed with viewing angle, looked at how the colors of the bird feather changed when you put it in a solvent instead of having it in air, having it underwater or in alcohol. And he found that if you put it in alcohol, it changes its color. And so he concluded that because the color depended on the surroundings of the refractive index, that the origin of the color must be physical or diffractive. Thinking a little bit about the size dependence and the angle dependence, inferred just from basically these macroscopic observations that the colors of the Indian roller must be due to some microscopic cavities, which he described, and he didn't know, he really discussed in the paper. It's a fun paper to read. It sort of speculated about all sorts of different shapes, but he concluded ultimately that they needed to be air cavities inside of some high refractive index matrix where the cavities were not small compared to the wavelength of light. Okay, so comparable to the wavelength of light.
Eric, can we make you move this way?
Yeah, no problem. I can still lean on this. I need to lean because I've been up for a long time. Yeah, so and it wasn't until sort of 76 years later that my colleague Richard Prum at Yale University sort of dug in and sort of looked at the nanostructure inside of this feather, both in real space and in Fourier space, to see what the structure is like. And in an electron microscope, the nanostructure that's responsible for this diffraction is not actually a bunch of isolated cavities. It's actually a bicontinuous structure, a disordered bicontinuous structure. But when you look at it in X-ray scattering, it's got a beautiful ring. So it's a disordered structure with a very well-defined length scale that's the same in all directions. This EM image looks a little bit anisotropic. In the early days, people weren't, we think that's just an artifact of cutting the sample open. So that in X-ray scattering where we don't cut it, we always see a nice isotropic diffraction pattern. So by looking at these, primarily the X-ray scattering and comparing it to the spectrographic data for over 200 species, Richard Prum and his very talented PhD student Vinod Saranathan, what they did is they looked at 200 plus species and they measured the peak spatial frequency that you see in X-ray scattering and compare that to the peak of the optical reflection spectrum. And they found a very strong correlation between the two, which made it very clear that actually these nanostructures which we're seeing are responsible for the color, and they were able to basically fit, not just measure a correlation coefficient, but basically fit the average refractive index of this structure. And because we know the refractive index of the protein beta carotene, they were able to determine that the volume fraction was about 34%. Yes?
Sorry, when we were thinking about superhydrophobic surfaces,
Yes.
we were sort of saying that the birds might do this to make them more water repellent.
Yeah.
And I just wondered how that fitted in, or am I looking at the wrong bit? Or?
Yeah, so that's super interesting. I'll give the short answer to that. So my understanding is that these porous structures, the primary drivers for their evolution were being lightweight but strong. So you want to have a porous structure to be lightweight and strong. You also want to have a porous structure to be water repellent, like the superhydrophobic surfaces. And the theory by my ornithologist colleague Rick Prum is that really these colors were naturally or accidentally stumbled upon on the way to the general evolutionary pressure was to be lightweight and superhydrophobic. So this is, and then all the crazy colors you see emerged from less obvious things about the positive feedback between traits and sexual preferences. So we can talk about that over coffee. So but there is a connection. There is definitely a connection to the water repellency. And like Julia, I am very happy to be interrupted to have questions, especially because I only arrived in India about 7 hours ago, and you might need to help me stay awake. Okay, so that's the Indian roller is not the only bird, right? And then that was this disordered network is not the only geometry you see in biology that produces color. So this is a bird from South America. This is the Cotinga mainana. It has a morphology that looks like more of like a bunch of monodisperse air bubbles, sort of packed together like a colloidal glass. And recently, Vinod, when he was working, he's now at CREA, but he was for a while in Singapore. He was literally looking out his office window in Singapore. And he said, oh, that's an interesting bird. And he got a feather of it, and he actually discovered that this bird from Singapore, its network structure is not a disordered network, but a very, very beautiful gyroid network, which is basically a trivalent three-dimensional network. So there's really a whole bunch of different morphologies within birds. And then, of course, birds are not the only things that can make amazing structures to produce color. Vinod also discovered that this butterfly from the United States actually has the same structure as this bird in Singapore. And then basically, Vinod did a ton of work and basically identified that basically any structure people have ever seen in soft matter, you can find in a bird feather or an insect wing. Now, the thing that's really amazing about this is that this material on the right is chitin. It's an extracellular matrix carbohydrate primarily with some protein in it. This structure, which is from a physics perspective identical, biologically arises in a completely different context. This is an intracellular thing that's made purely out of protein. So there's two completely different biological and chemical pathways and evolutionary pathways for finding this same amazing, highly ordered structure. Now, so the big question is here, as soft matter scientists, do we know how to make these structures, okay? And in some cases, it's easy to mimic the structures that we see in living organisms. So this structure that Rick found in a South American bird was very easy for us to recapitulate with like colloid science. The basic idea is that we could make this structure through self-assembly, and self-assembly, of course, is the process where you start with some building block, and then you concentrate it, you equilibrate it, and you let it find its ground state in sort of a thermodynamic sense, or a near ground state. And we were able to do that with colloidal particles to make sure that the ground state was disordered. We used a bidisperse mixture of colloids with just the right size ratio, and we could make structures that look very much like what we saw in the birds and have very similar color. So that was easy. We could do that. my PhD student could do it in about 6 months, okay? The thing that is hard is something like this, okay? So this is not, you can't assemble this from colloids, at least without conventional colloids, because it's a continuous network, and it's a bicontinuous network. There's no single building block that I can assemble. I mean, maybe I could have little trivalent things, but getting together match so smoothly would be very difficult. And you know, this is a really beautiful structure. This is sort of a 3D rendering of what the single gyroid looks like. And there's beautiful long-range order in these things. So you can have hundreds of lattice constants in all 3 spatial dimensions. So these are real proper, beautiful crystals at photonic scales. Now, can we do this with soft matter science? Yeah, Sriram.
I mean, when you say can we self-assemble, you're not worrying about the fact that, I don't know, copolymer mesophases have many of these things.
Yeah, here we go. No, you can't do it. You cannot make this structure with block copolymer. So the closest you can do is with a tri-block, you can concentrate it and equilibrate it using the paradigm of self-assembly, and you can get a so-called double gyroid, which is an interpenetrating network of 2 of these guys. You cannot directly assemble the single gyroid, okay? The second point, as far as I know, the best you can do is remove one of the networks after the fact to make the single gyroid. You can't directly assemble it.
Eric, is the red one rendering of the bird data, or it's just?
Oh, no, that's just the mathematical model.
Picture.
Yeah. No, we don't have beautiful 3D data like that. But all the X-ray scattering and the EM points to that being a very good representation. Yep. And wait, let me just finish this last point, and then I'm happy to take another question. So, and not only can you not make a single gyroid from block copolymers, it's also completely at the wrong length scale. So the key thing when you're doing self-assembly is that the spacing of the structure is always linked to the size of the building block. And so using conventional chemical routes that have been developed so far, the best you can do is you can make a double gyroid at length scales of tens of nanometers, a whole factor of 10 away from the length scale you need to be optically active. Okay, so there's a problem with size and there's a problem with symmetry. And I think Sayantan had a question.
Yes, so I was wondering that this black portion of the feather, like it so the structure is different, right? Yellow and black, both are like.
Oh, thank you. Oh, you mean
Yes,
macroscopically, not macroscopically, yeah. So the bird actually, you know, it's amazing what they can do, right? So at different feathers on the body, they actually can have different nanostructures. And I believe, if I remember correctly from my discussions with Vinod, that these distinctions in the bluish hue, the yellowish hue, and the greenish hue correspond to subtle differences in the lattice constant of this thing, which is what makes one of my points. And then the black section is more driven by pigmentary interactions. So birds have both the structural physical axis as well as the chemical axis, and they interact with each other. And they have, and this pattern of yellow, green, and blue is not random. You know, every bird like this in Singapore, unfortunately, I don't remember its name, has that same pattern, right? They can precisely control the lattice spacing at these very large length scales. Yeah.
So comment, this is a leaf bird, and you can find it here.
Oh, great. I thought you said you weren't a birder.
No, he is.
I know, but when Bobo pointed out that you were a birder, you shook your head. Yeah.
You won't find roller here.
I won't find the roller here. Okay. I was hoping that I would. I wonder where Raman saw it, though. Okay, he didn't say where he got the bird feathers from. Yeah.
In the case of bird, how does the pattern arise? Is it like some Turing-like mechanism?
Oh, the macroscale?
Yeah.
Macroscale would be questioned for another time. I don't know the answer. I have enough problems at the microscale.
No, I'm talking about the microscale.
Oh, we'll discuss. That's precisely. We'll discuss one of my hypotheses about how they do that for the rest of the talk, basically. Yeah. Mm-hmm. That one is it's around 10 nanometers. Yeah. Yeah, in the back. Yeah.
So why is it so? Like these are the SEM images of the birds which SEM images are these? These are the
Yeah,
SEM
so these
images?
SEM images correspond to the feathers, the colored feathers, or the colored scale, wing scales.
Okay, so these are also exhibit the properties of the metamaterial? Or
Yeah,
just-
I call it a metamaterial. It depends on what your definition of a metamaterial is. The point, when I think of a metamaterial, is that macroscopically you have a material whose properties is determined by structure at the mesoscale, not at the molecular scale. And compositionally, these two birds are made exactly out of the same molecules. They just have different mesoscale structure, and they have completely different macroscopic properties, and that's why I call it a metamaterial.
Okay, so if you want to replicate such kind of periodic structure
Yeah.
in the labs,
Yeah.
will it exhibit the property of metamaterial?
It'll have the metamaterial property that I just described, but it might not have all the fancy, polarization and other types of things.
Okay.
I'm only referring or negative index, all that stuff. I'm just referring to a more general definition of metamaterial. This might be like the turbulence point. Yeah, I'm calling it metamaterials just because the macroscopic properties are not determined simply by what it's made of, but by mesoscale structure.
Okay, yeah, thank you.
Maybe you're talking about that, but you can have, with a surfactant, you can have a charged surfactant, you can have bicontinuous phase, which are space enough, right?
Yes, absolutely. With charged surfactants, you can make structures that look very similar to, I think you can even get
You
the
can get that,
single
yeah.
gyroid
And you
with
can
it.
have the disordered one as well. So
Yes,
that's-
exactly. Though, interestingly, as far as my biologist friends tell me, there is no membrane involved in this one. So, but yeah, no, that's an excellent point. Yeah, no, thank you. No, thank you. I was pushing the block copolymers, but I left out the amphiphilic phases, which can do much better job. The only issue there with the amphiphilic phase would be that they're very fragile, right, compared to the bi-block copolymers. But certainly you can get similar symmetries from the amphiphilic phases. Okay, so I look at this and I see that there's something missing in our community of the way we think about assembling structures in soft matter. And I think the two main things here are that we want to be able to systematically be able to control the structure of materials at scales much bigger than the building block. And the other thing we want to be able to do is we would love to be able to get a whole range of different sizes and symmetries and topologies from the same building block. And there is something we've known about in soft matter for a very, very, very long time, since before soft matter existed, where we can easily get structures at scales much bigger than the building block, and we can get all different sizes from the same building block. And that framework is condensation or phase separation, okay? So to remind you of what that looks like is that microscopically, the simplest case is to think of having a mixture of blue and yellow things. And if I zoom out macroscopically, blue and yellow make green. So green denotes that I have a homogeneous mixture. And if I increase the attractive forces between like particles or increase the repulsive particles between different particles, then I can microscopically want things to be like similar things. And I can macroscopically separate it into domains of one composition that's distinct from the other composition. And of course, because this is thermodynamic, I can go back and forth and back and forth. This thermodynamics has been known for forever, as far as soft matter exists. And the phase diagram looks like this. So this is temperature or interaction parameter on the vertical axis. And the horizontal axis is, you can either think of it as your order parameter or your volume fraction of one of the components. And the key point is above some critical temperature or below some critical interaction parameter, your system remains well mixed, like that. But if I take the system and I quench it into this shaded region, it will spontaneously separate into two phases at either end of the excluded region. So this is what thermodynamics tells us. But thermodynamics only tells us what the composition of the two phases is and how much of each phase we should have. It says 0 about what the structure should be. And typically in classical phase separation in liquids, once you've made the right amount of each phase with the same composition, interfacial energy kicks in, and interfacial energy, of course, makes things that are non-spherical tend to be spherical, and it also tends to make small things bigger. And so if we look at this process, this classical process, in real life, what does it look like? So here is a mixture of silicone oil and fluorinated oil. This, we'll be looking at this system a lot in this talk, where we're starting off in the mixed region, and then we're cooling it down and we're quenching it. And you can see the nucleation and growth of the oil, the fluorinated oil-rich phase inside of the silicone oil-rich phase. And you can see how interfacial tension, once you've created these phases, drives everything to coarsen. Now, the thing that's really great about this process is that inherently it samples every single size that you could ever want, from the molecular scale to the macroscopic. Coarsening spans every length scale, which is amazing, okay? But the problem is that none of those length scales are stable, right? So the question is, the hypothesis that we put forward several years ago is that maybe, because we know, and that's a talk for a whole nother situation, we've learned recently that cells are really good at controlling phase separation, right? So we have this hypothesis is that maybe biological cells start with something that looks like classical phase separation and then add another term to the Hamiltonian to select a length scale, okay? Or to prevent coarsening. And so our idea was to take a process that looks like this, which is conventional phase separation, and embed it in a network, okay? And our initial idea came from looking at cells, because we know that cells have a polymer network in them called the cytoskeleton, and we thought maybe the cytoskeleton might have played a regulatory role in selecting length scales. And I saw a question in the back, yeah? Yeah. Maybe you can take the mic and ask again. Thanks.
Yeah, it depends upon up to what temperature you are bringing it.
So what part? So the size, or?
Yeah, size of the.
So in this example, like if you're above the phase boundary, you remain mixed. If you're below the phase boundary and you separate, it really wants to be macroscopically phase separated. If you wait long enough, you will just have one big droplet inside of another.
So as long as you are in two phase, the sizes will be remain same, irrespective of the temperature.
It will always go to macroscopic. It will always be one big bubble inside of the other. The ultimate sizes are set by the dimensions of your container. So yeah. Okay. So the idea here is if we have the same process of phase separation, but now if it's hosted inside of a polymer matrix, then that could maybe add a restoring force that opposes coarsening. So in this schematic, we show the basic concept. We imagine that the discontinuous phase, in this case, the droplet rejects the polymer network. So in order to grow, it needs to deform the polymer network. So as it grows, as it coarsens, you dump elastic energy into the network, okay? And that can create and our hypothesis can create a counterforce to surface tension, which makes things want to coarsen. Okay, and the talk is primarily about exploring this hypothesis. Okay, so how do we do it? We start with a piece of silicone gel. So we love silicone because it's transparent, and we can very easily, with the same system, tune the stiffness by changing the ratio of crosslinker to polymer. And we take that gel and we put it in a bath of fluorinated oil. And we just let it swell at whatever temperature we want for several days to let everything diffuse where it reaches its equilibrium. So I've now shown the inside as being green instead of yellow because it's homogeneous mixture of the oil and the and the polymer. And then we cool it down relatively quickly with a controlled rate, and we let phase separation happen. Okay, this is what it looks like after several days of equilibrating. Look, this is a scale bar of 100 microns. we look at it in the light microscope, and we beautifully see absolutely nothing. And if you're an experimentalist, you might appreciate how hard it is to actually have a sample be that clean over lots of microns. So this is a super homogeneous mixture. I was very happy to see such beautiful nothingness. And then you start cooling it down. The timestamp is up here. And you can see, just like before, we get localized nucleation and growth. But now there's no coarsening. The coarsening has completely disappeared. So, so there's something interesting here. And if you look at, we know that the coarsening went away because we added network stiffness. If we compare the size of the droplets in a soft network to a stiff network, the intuitive thing happens is that the stiffer, in the softer networks, the droplets are larger than in the stiffer networks. And that makes sense, right? Because in the stiffer networks, the elastic network is pushing harder against the droplets and resisting the coarsening more. Quite nicely, these droplets are very monodispersed. So these are size distributions of droplet radii for stiffnesses from 800 kilopascal to about 4 kilopascal. They're all monomodal. And they get a little bit fatter as you get to larger particles, but actually if you normalize the width by the average, then they basically have the same like 8% or so monodispersity. So these are quite monodispersed droplets, which is interesting in its own right, actually. But we won't talk about that so much. And so yeah, so so this basic hypothesis seems to work, and a great place for questions. Yeah.
How are you getting the elasticity values for all these different particles?
Oh yeah, we just measure them. So in practice, we vary the ratio of the crosslink. So the polymer can make crosslinks at its ends, and then we can add crosslinkers that are multivalent. And as we add more and more crosslinker, we have fewer and fewer free ends, and we even have some free chains at the really low crosslinking. And that makes a denser and denser network, makes a stiffer material, and then we just poke it macroscopically to measure Young's modulus.
Poke it with what?
So we've done it in a couple of different ways. For the stiff samples, we can do tensile testing. And for the softer samples, the tensile testing is hard. So we put it in a dish and we poke it with a cylindrical indenter.
Just one more question. So the clean sample from where you're getting the different size particles in the previous slide.
Yeah.
So these are the same samples, right? So
yeah, these are the same samples.
So how do you isolate these and then take it to a different probing mechanism?
Sorry. These stiffnesses I report here are the stiffness of the material before I swelled it. It's the pure polymer before I process it. Yeah, lots of questions. Great. Yeah, I'll let the microphone people decide.
A quick question. Yeah. So I think it's quite intriguing that the polydispersity index doesn't quite change.
Yeah.
What sets that number 8%?
We do not know.
So do you think this is kinetically arrested state, or it's a true, in the sense if I were to take two drops with tweezers and merge them, will they merge? Or?
So I can't, because the elastic forces are so strong, the tweezers can't do anything. For this case of droplets, I don't know. I do know in the case here that the final size you see is dependent on the cooling rate. Okay. It depends logarithmically on the cooling rate.
Okay.
I have no idea why, but it does depend logarithmically on the cooling rate. The results will show you later with things that look more like birds. It's really thermodynamic and totally independent of the cooling rate. Okay. And we might get there. Yeah.
Yeah, I was just wondering. I mean, if you think there's something like a Laplace pressure of the drop is being balanced by some modulus of the medium, you might expect an inverse proportionality to the size, and that didn't seem to be quite the factor.
No,
I mean, like 840 to 4.2 is some factor of 5.
We have a factor of 3 over 3 orders of magnitude. So yeah, that was our, actually, as you may know, some of you may know, I love this field called elastocapillarity, and like our whole hypothesis going into this was that exactly that's would be what would happen, and that's...
And it's not quite,
it's not quite, yeah.
So in the picture that you had in the previous slide.
Yeah.
Yeah. So once you have the first drops nucleating,
Yeah.
you start building up elastic stresses.
Yeah.
Does it also set where the next drop is going?
Beautiful. Yeah, no, absolutely beautiful. So you can measure G of R for this 3D thing, and you see that G of R monotonically decreases as the things go close to each other. So basically, G of R equals 1, like greater than the spacing, and it sort of slowly decays to 0 as you come to contact. What's really interesting is we don't know. There's two ways that that could happen. One is because of the elasticity, or it could be because of diffusion. So when one droplet is growing, it's eating up the stuff around it, and so they could also be long-range coupling, either diffusively or elastically. So I know there are lots of other questions, but I'm going to move forward, and I promise to take more questions. So yeah, so one important thing is most droplets, you know, their shapes are governed by surface tension, right? So as I mentioned, here we also saw droplets, all of the droplets we looked at were spherical. The question is, are our droplets spherical because of surface tension? It seems obvious that they would be, but we were careful, and we did this following experiment. We made our sample, we stretched it, we swelled it in the stretch state, and then when we cool it down, if surface tension or interfacial energy was the boss, we would get spherical droplets. But if elasticity was important, it would break the symmetry and our droplets wouldn't be spherical. And of course, I wouldn't be saying it unless it was the case that when you grow these droplets, you can see that as soon as they're resolvable, like, you know, compared to the microscope, they have this non-spherical shape. So that somehow the growth of these droplets is not just feeling the modulus, it's also feeling the state of stress in the material. So there's some, so surface tension still must be important somehow, but it's not what purely determines the shape of the droplet. So yeah, so that was the first half, and you know, I'll say that we have lots of hypotheses, but we have very few answers about what we just talked about. Generally, the key point here is that when you have droplet growth in a matrix, the mechanics of the matrix governs the size. It can govern the shape. It can also govern the stability, which I didn't have time to talk about, but I'll talk about the same phenomena in the second half of the talk. Now, the question here is that these were beautiful and interesting physics, but you know, having isolated droplets in a matrix are not very useful and don't match our original motivation for the talk, right? We were really motivated by these beautiful, strongly correlated, organized structures inside of bird feathers. And so there's two things if we want to have things that look more like bird feathers. We need to have stronger structural correlations. We need a G of R that has a peak, not just a decay. And we also need smaller features. These things have, look at the 100 micron scale bar versus the half micron scale bar. We need to shrink this all down to get bird feathers. So there's two, you know, basic hypotheses to get structural correlations. Just like any other soft matter, we need more droplets. They need to interact with each other if we want them to correlate. And we probably need stiffer matrices to get smaller features. So this is when Carla came to my lab. And you know, unfortunately, I'm a physicist, and so I don't understand chemistry. And Carla came to my lab as a physical chemist, and so she understands physics and also chemistry. And you know, we were looking at the phase diagram of the material of the previous section, and we'll see a lot of these phase diagrams. So I'll spend a moment explaining it. So we have incubation temperature, the temperature at which we swell the material in the bath, versus how much liquid gets sucked into it. So this is the amount of swelling in the liquid. And you can see for the oil that we were using, even if we go over a big range, we get very little amount of oil coming into it. It's hard to imagine a high volume fraction when you have that little oil. And because she has great chemical intuition, she's like, well, you know, silicone's not going to like all these fluorines, so let's get something that looks more like a hydrocarbon and increase the solubility. And this is how much the silicone can swell in this new material. And what's really important here is that imagine that I'm swelling the system at this temperature and then cooling it down. When I cool from here, I only get a small difference in the solubility, so I only get a few dilute droplets. But if I cool here, there's a huge difference in the solubility, so I should be able to get more higher volume fractions and get structures that are correlated. And nearly on the first try, she got that, this is one of the very, very first images that she took in the lab. And this is an unprocessed image. It looks a little bit like a mess. And this is moving the microscope. This is regular imaging in a bright field microscope, moving up and down. It doesn't look like total random garbage. It looks like there's some structure in there. If you apply a filter and you threshold it, you have this beautiful bicontinuous structure. Okay, and it's really bicontinuous. And if we Fourier transform, not this, that would be cheating to Fourier transform that. If we Fourier transform this, we get a nice peak at a well-defined length scale, like we see in the bird feathers. So here's a bird not from India, but a bird from my backyard in New York. This is what the X-ray scattering spectrum looks like. Okay, so qualitatively, it's pretty close to what we're seeing in a bird feather, though there's two big differences. One, the peak is at larger length scales, and of course it is, because if it was like the bird feather, I wouldn't be able to resolve it in my light microscope. And the other feature is that the delta Q over Q is fatter here than here. But what I don't know is I did this by doing a Fourier transform of an image in a light microscope. Maybe it's as narrow as this if I did a proper scattering experiment. So maybe it's not as good. It's certainly too big. But nevertheless, we can try to figure out what's going on. These networks are truly elastic. There's no creep. They're rubber. It's rubber, basically. Yeah, so this structure, you can see structures like this in fluids, right? So if you take a fluid and you quench it through the critical point or near the critical point, of course you can see transiently, you can see similar channel-like structures that eventually coarse in and increase their contrast. So here we're cooling down something near the critical point of an uncross-linked mixture of PDMS and liquid. Early on, just below the critical point, we see this nice channel. It coarsens very quickly, and then it turns into droplets, okay? But now I'll show you the movie of what happens when there's cross-linking. We're mixed, we're cooling down, we get the structure, and it doesn't coarsen. So what you're seeing, there's a drift in the microscope. We're changing the temperature of a sample in the microscope change by 40 degrees C. So there's lots of thermal expansion, and the jumping that you see, occasionally there's a shut-in shift. That's Carla adjusting the focus on the microscope. But you can see by eye, and I'll show you in a moment, quantitatively that the length scale isn't really changing. This is what the pictures look like, still images. Okay, so yeah, again, we can look at how the morphology changes with the network stiffness. And as you would sort of expect that as we, from the previous section, if we have a relatively stiff network, this is nearly a megapascal, has a length scale around just under a micron. And as we go to softer and softer networks, more compliant networks, our network has a spacing more like 2 microns. If we make the network too soft, though, we break the network. I might show this data later. I don't think I have time to show it, but if you ask, I can show you. These initially form through the same sort of channel-like structure, but then eventually they turn into droplets. But they stop growing eventually, and it looks like before, except the droplets are close to each other. And if we do Fourier transforms of all of these, we see a scaling, and this sort of answers Sriram's question, I think, quite clearly. The peak length scale in the Fourier transform versus the modulus, if it was elastocapillarity, would scale to the -1 power, but this scales to the -1/2 power.
And that's true even your bottom images?
Even if the bottom images, which is really shocking, actually. To the point that I really didn't believe Carla at all. She needed to really convince me by showing it over and over again that, and the scaling looks like it might be turning over at really low stiffness, but it seems pretty continuous though, actually. And just like before, if I do the phase separation in a stretched sample, the microstructure is sensitive to the state of stress. So here is a Fourier transform of, the imaging here is lousy because it's really hard to do high magnification imaging on a stretcher. So these images are not as good to resolve by eye, but if you do the Fourier transform, it's clear that you get anisotropy. And if you manipulate this image by doing a bandpass filtering and an image gradient, you can see clearly that the microstructures also become anisotropic. So again, suggesting that there's a direct coupling between morphology and stress. So now, okay, we don't know what's going on, and now I'm going to go through and try to do all the things that we would do normally for a phase transition and ask, does this look like a phase transition we recognize? So the first thing we need to say is that this is a continuous phase transition. It's not a first order transition. Looks like a second order transition. First, if you just look at these images at different time points as we're cooling down, what you can see is the length scale doesn't seem to change very much. It's just the contrast that appears to increase. If you're not good at doing Fourier transforms by eye, luckily we did the Fourier transform for you. And so here's a series of Fourier transforms at different extents of cooling. So as we go this way, we're cooling the sample deeper and deeper. And you can see as we cool, the peak of the Fourier transform doesn't shift. It stays in the same location. This vertical line is in the wrong spot. I'm sorry about that. It should be shifted to the left a little bit. But the point is that we can look at this peak, we can fit it, we can get a characteristic of length scale. So the transition is continuous. The other thing about the transition, which really to me was shocking, is that it's hysteresis free. Okay? So if I take the height of this peak and I call that the contrast, okay, and I start off hot and mixed, I cool down, I get here, I start seeing some structure, the contrast increases, and then we heat it up, it follows exactly the same path, okay? This is not what you would have expected if you were off critical spinodal decomposition. Off critical spinodal decomposition, when you cross the spinodal, you would expect it to be a continuous transition, but you wouldn't redissolve until you hit the binodal, and then you would have hysteresis. So that really surprised us. And also the length scale is also hysteresis free and continuous. Yeah, it's 2 pi over Qmax, yeah. Yes, Sridhar.
But you're not necessarily saying there's a phase transition here at all, right? Because if I had a bunch of hard spheres, let's say, and I just went from low concentrations, I took a bunch of hard spheres and went from low concentrations where it is like a vapor to high concentrations where it is more like a liquid, you will start to see the first peak of the liquid state structure factor at some point.
Yeah.
There's no phase transition there. It's just, low density to high density.
Okay, you will have to explain that to me later.
Yeah, I mean, meaning if you take a system, take a boring equilibrium fluid medium with no attractive interactions, and just study it as a function of the number density of particles.
Yes.
At some point, the static structure factor of the particle number density will start to show a structure peak.
Yes.
The place where you go from no structure peak to structure peak isn't by any means a phase transition.
So sorry. I understand what you're saying, and I think, but that's when I'm calculating S of Q at the single particle level.
Collective density.
This length scale is about 2 orders of magnitude bigger than the size of the molecules. This is not the
that's
peak
okay.
in
I'm
S of
talking about
Q.
droplet. You've got some effective little things, self-assembled little blobs, and you're packing them. If you pack them more density, you will start to get a structure peak. And if they're rarefied, you won't.
This sounds like we're getting to the heart of the question. Okay. I would still say that the difference here is that the blobs don't exist until I start cooling. Right, so
that's an interesting point.
It's also another, and maybe it's not a phase transition. It smells like one to me, but I'm open-minded here. But I'm going to try to get through the data so you can know the full story. So the other thing is that the lengths, there is no coarsening, like I said. So if I measure this wavelength and I keep the temperature fixed as a function of time, we see no coarsening whatsoever. Okay, it's completely constant. And also very, very importantly, which contrasts it to many other known mechanisms for making arrested bicontinuous structures, the wavelength is independent of the quench rate over about a factor of 30, okay? So we don't really see any dependence of this length scale on the quench rate. So that doesn't smell like what people call arrested spindle decomposition. Okay, super. Yeah, and so another interesting point here is that if you have a stiffer matrix, you have to quench deeper to see a structure. So look on the right here. So we're just measuring that length scale as a function of how much undercooling we did. So on the right is we equilibrate everything at 60 degrees. If we take a 0 kilopascal sample, a liquid mixture, as soon as we cool the tiniest bit below 60 degrees, we start to see phase separation. We can measure a length scale. And as we keep cooling at our finite rate, we see that length scale get bigger and bigger. If we take intermediate stiffness one that ultimately makes droplets, we have to undercool it a little bit more, like a few degrees before we see anything at all. When we first undercool it, it has this network structure. I can show you the rest of the data later. I don't have time to show all the data. But at first, it has a stable length scale, and the contrast just increases. And then it starts to coarsen as we cool deeper and deeper. But for these guys that really have the beautiful network structures with the stiff samples, we see absolutely nothing at all until we undercool about 15 degrees Celsius. So the system remains under the microscope perfectly mixed. And then we start to see this peak that's constant and independent of how deep and how fast we cool and perfectly reversible. All of this is, yeah, it seems very thermodynamic to me. So we tried putting these things on a phase diagram. So here's an example of a phase diagram that corresponds to our stiff samples that makes the network bicontinuous structure. And there's 3 different lines on this phase diagram. The white circles are analogous to the ones I showed earlier. This is if I take a slab of the stuff and I put it in an oil at a certain temperature. At 60 degrees, how much oil do I take up? So at 60 degrees, the rubber swells so that 55% of it is liquid, okay? So this is really the thermodynamic coexistence. This is sort of like the Flory-Rayner equilibrium of a gel and its solvent, okay? So this is definitely equilibrium thermodynamics. And then if we can start off at different temperatures, and these 2 dots, the circles are showing us where did the nanostructure appear upon cooling, and the crosses show us where did the nanostructure disappear upon heating, and those 2 lie on top of each other, okay? The gap, so here there's coexistence of 2 unstructured phases, one liquid, one solid. Down here we have coexistence of sort of a macroscopic liquid with a microstructured elastic thing, and in between we're calling this the mechanically stabilized mixture. And if we go to a softer sample, this is the softest sample that still has the same network bicontinuous structure. It has the same basic structure, except now this gap between of the mechanically stabilized state gets smaller and smaller. So thermodynamically, I want to point out here that what I said earlier, the transition to the microstructured state is continuous and free of hysteresis. That sounds like we're quenching through a critical point in a normal phase diagram. However, we see that same phenomena everywhere on this line. So this thing looks like a critical line of some sort. But I'm not even sure if I'm using the right words. This is all just phenomenological. I have a line of points when I go through them act like a critical point. And at least to the amount of thermodynamics I know, I don't know how to map this onto a phase diagram that one would typically see in phase separation. And if anybody has ideas, I'd love to hear them. And so the key points, we'll have lots of time for discussion, is that living systems, are really good at patterning things much larger than their building block. And I think there's I should have said unexplored mechanisms of control. Biology is really good at controlling phase separation, and I think we can learn from them. Even though we didn't work in a biological system, we were inspired by elements of biology. In our case, where we hypothesized that it was the cytoskeleton or the matrix that confers elasticity, that matrix basically changes everything about phase separation. It changes the size of the objects, the shape and the stability of the objects. And as I said on the last slide, this process seems to follow sort of an unusual pathway. To what are we doing now? One thing we're doing is because the oil that we're swelling things is volatile, the structures over long term are not stable. So we can polymerize. The oil that we use, we can also polymerize to stabilize things. Also, if you look at these structures macroscopically, you can see that they look different. You know, the ones that with structures that look similar to a bird feather are starting to make color. We want to understand that and control that and make lots of different things. We're also looking at mechanical properties. So this is a fun little, just a teaser. If we just measure the stress strain curve of the rubber that we start off with, it looks like this. If we polymerize the oil and have just the pure polymer, the glassy, pure glassy polymer that we're polymerizing inside of the network, it has a stress strain curve that looks like this. It's much, much stiffer, but it's super duper brittle. But if we polymerize this bicontinuous structure, we have a strain curve that looks like this. So it increases the modulus compared to the elastomer by a lot, but it also increases the toughness. So this material, it can sustain a lot more damage before it breaks. So there's interesting mechanical properties. And we're trying to figure out, you know, if we can do this with different types of materials, and I'll leave it at that. And I'd love to hear more of your questions and your suggestions. Thank you. I think Sayantan had his hand up first, or at least I saw you first.
Yeah. When you said that the droplet shape, it tracks the stress when you did polymerize and the stress. So I was wondering that, have you looked at the local orientation of the polymers, like whether it is the stress or the local orientation?
Yeah, that's a super, super question. I will tell you what we've tried and what we've seen. Sorry, what we've tried and what we've seen and what we think. Okay, so you know, if you take a macroscopic piece of PDMS and you stretch it, you can rotate the polarization and you put it through cross polarizers. You can see this really beautifully. But the problem is these structures are very, very small, and so the birefringence is not very strong. So in normal cross polarizers, we can't see everything. We've also tried a pole scope, and we can't see anything in a pole scope. Maybe with real experts of the pole scope, we might be able to see some orientation near the droplets. But I would say what I expect to see there is that if the droplet, well, we do know from separate measurements that I didn't show, is that when the droplet is growing, if it's excluding the polymer network. And so I expect the polymer network to be aligned parallel, tangent to the surface, is what I expect. And that would be slightly different when you nucleate before you stretch before you nucleate. Mm-hmm.
Thanks.
Yeah, microphone.
So have you looked at the kinetics of coating, in the sense that if you look at the droplet size up to the point where it stabilizes?
Yeah. So the for the droplet case, right? So we looked at the kinetics super carefully for the network, the bicontinuous case, right? And we saw nothing. There was never, you know, as whenever we saw a structure, it was always the same size. For the droplets, you saw it nucleates and it grows. And we have only done very cursory explorations of that trajectory over time. And
I was wondering if it depends on elasticity.
Yeah, no, that's a great point. I would say that we should probably do more of that.
So your mechanical, the fact that your mechanical properties are somewhere in between these two things.
Yes.
There's some work by this guy in Hong Kong who embedded, I think, microspheres in PDMS and controlled the G prime of the PDMS.
My former postdoc Chin.
Or Ichiu.
Yeah.
Ichiu Zhang. Yes.
Oh no, wait! I thought it was Chin Zhu, my former postdoc.
Maybe both of them.
Okay, maybe they're on the same paper, and one's the experimentalist and one's the theorist.
Yeah.
So anyway, finish your question. Sorry.
Ichiu is an experimentalist too, from Bob's lab.
Okay.
I think they were together. I met them at.
Okay, maybe.
But you know that scaling, right? So they actually varied the G prime of the embedding medium and showed this beautiful scaling of the.
That's great. We should compare to that. Right now, we only have that one curve that I showed you, so we could fit it to any scaling that we'd like.
So on the mechanical property, so if you make any mechanical experiments, do you keep the same structure factor after that, or do you get any evolution of?
Does the morphology change when we stretch it?
Stretch or do I don't know.
Yeah, that's a great question. So on the, you can see with this experiment, it's not. This thing is not amenable to sort of doing cyclic stress, but I think that would be a great experiment.
Because when you show that at least the mechanical property are completely different from the elastomer alone.
Yes.
What type of experiment did you do for that?
Yeah, so this is a
yeah.
macroscopic tensile test. That one we can go back and forth, and there we see strong hysteresis.
Okay.
So it looks a lot like the Mullins effect.
So then,
It looks a lot like the Mullins effect, yeah. There's a hand in front.
This phase transition that you mentioned, have
Yeah.
you thought about what would be the auto parameter that would grow at that phase transition?
Yeah, I mean, I would just say that my guess is just the volume fraction of the oil phase, but there might be something smarter than that.
There's no symmetry.
Yeah.
So it's like liquid gas.
If you can define another parameter, then it's okay. And it looks like the contrast looks very much like another parameter.
Yes, okay, so you think it's a reasonable order parameter. I think we had a question up front, yeah?
I was just
Oh, there's a microphone.
I was just wondering if you characterized the 3D structure more carefully, like is it a gyroid or is it a mixture of many things?
Thank you.
How
Yeah.
you do that?
So yeah, we do have some pretty nice confocal images, and it really it's funny, a referee just asked us that same question. Anyway, yeah, so, and we did sort of a topological analysis of like the connectivity, and the first thing to say is that because of these structures are right at the edge of what we can resolve with the confocal, I'm not sure that I trust our topological analysis, but we do seem to see a wide mixture of like connectivities. The junctions are not always threefold or always fourfold. And it's really a disordered network, yeah, both in topologically and translationally, yeah.
Thank you.
I'm just going to do what the microphones do, yeah.
Yeah, so for the barb feather, if I look at the feather, I think there is a mid stem, and then there are some fiber-like structure on the sides.
Yeah.
So, and then those fibers are also arranged in some order. So, so that length scale is much larger than what?
So the main shaft of the feather is called the rachis, and then the things coming out, the next generation of the barbs, and the generation after that are the barbules. So all the colors here are on the barbs, and the centers of the barbs are primarily air, and there's basically one layer of cells around the outside, and all the structures are in that one layer of cells around the cylinder of the barb.
Okay.
Okay. The cell and the cells are, so this is much smaller than that, of course,
Okay. And another, if this is a second order kind of phase transition, so if you measure specific heat, possibly there will be some signature.
Yeah, great point. Yeah, so that's an excellent point. Thank you for reminding. We tried doing some calorimetric measurements in the early days when we just had drops, but it was really hard because there was such a small volume fraction. But we should totally try it again now because we sort of have 50-50 volume fraction. There might be a bigger signal, and we might be able to do something calorimetric. Thank you for that suggestion. That's a great. We need to revisit that. Yeah.
Hi.
Yep.
So when you are taking the Fourier transform of the images, I can see the Q value going beyond 0.01, which is like less than 100 nanometer. So how is it possible to resolve?
That's not resolved. That's just the pixel size at the end of the day. At some point, as you go to the right, things stop being resolved. But you can still Fourier transform whatever pixels you have on your camera. So.
Now, I know that in lyotropic gyroid systems, they say it's by just measuring the conductivity, for example. Can you do something like that?
Yeah, so of course, now that I moved from Switzerland to the United States, I have to find ways to get money. In Switzerland, it was easy to get money. In the US, not so much. And this is, and when you think about potential applications, in addition to sort of the optical and mechanical properties, really interesting idea is to use this to make different types of like porous materials, either for batteries or for filtration. And that is really something we're going to try. We're going to ask for money to try. And the problem we just need to, again, we have chemistry problems because I'm just a physicist. We need to figure out how to remove one of those phases if it's going to be a fluid flow. The other thing we're looking is making one of the phases conductive so that we could establish it with conductivity. But that's exactly where the money is, I think.
Yeah, so I was just wondering that as you said that droplet size can be controlled by the stiffness of the polymer network. So if you keep the stiffness of the polymer network same and change the interfacial tension between the two phases, will it be the same effect or will it be some?
That's a great question. The closest we've done to that is we have, I'll just leave it at that. I don't know yet. Yeah, so the best way to do that would be to try different oils and different mixtures, and measure the surface tension between the two phases before in the uncross-linked case, and then try to correlate size to that. I think that's a great idea. We haven't done it, so yeah.
Just a quick announcement. Who hasn't registered yet? So the registration material is like if you go this way and take left, the last room you have the registration material. So please collect your registration material over the lunch time. Lunch will be in Astro Terrace.
So the phase diagram is
right. Yes, sorry.
concentration and temperature, and that's where we saw the line.
ICTS, which is a neighboring institute just a few kilometers from here. He will chair this session.
Thank you, Pramod. Welcome back, everyone, to the afternoon session. The first talk is by Prerna Sharma, who is at an institute which is much closer than mine, by the way. So she'll talk about enhanced mixing and vertical flow around confined microalgae.
Yeah, sure. Sorry, I'm nervous. Okay, so I'm going to talk to you. Where is the pointer? Pointer is not on. Okay. Yeah, thank you. I'm an experimentalist. I should be able to figure this out, but I'm extremely nervous. I'm going to talk to you about enhanced mixing and vertical flow around confined microalgae. Most of my co-workers are actually here. Some are not. This is work of two of my talented graduate students, Debasmita and Sujit. I'm not sure if I'll get to Sujit's work, but if time permits, I will showcase his work as well. What you're seeing on the right is single-cell algae called Chlamydomonas, and these beautiful colored lines are trajectories of tracer particles around it. So you see these vertical flows all around, and they arise when the algae is confined. So this talk is aimed at graduate students and so on. So I'll spend some time introducing what this organism is, why is it interesting, etc. So the outline of my talk is that I will talk about this model system of Chlamydomonas reinhardti. Then I'll talk about how their flow fields and motility changes when you confine them. And if time permits, I'll talk about the phenomena of phototaxis of these cells and the collective effects that are present in it, and conclude. So algae in general is a very interesting system to look at. It's because it has major ecological implications. So for example, you can have these large scale pattern formation by algae called algal bloom, and they can be toxic to the environment. So you really want to avoid this kind of growth. It happens due to pollution in sea and so on. And then, I'm sorry. We also use algae in industrial bioreactors to purify important things like pharmacological drugs and dyes and other important molecules from their cell bodies. So, Chlamydomonas reinhardti is like a simple harmonic oscillator of biology. It has these two flagella that beat in this beautiful breaststroke manner. This cell is held on a glass micropipette, so that's why it's not swimming away. If it were free, it will just swim away like this. The flagellar beat frequency is 50 Hertz and the cell speed is about 100 microns per second. The cell size is about 10 microns. So, it's a very popular model system mainly because it's very easy to grow in the lab and there is a large collection of mutants available in international repositories. So, in an unbounded medium, the flow field for such organisms, and actually in particular for clammy, is what is called Puller type, where on a beat averaged time scale and on large length scales, you will see fluid coming in from the front and back of the cell and being pushed out from the sides of the cell. And so that's why it's called a Puller type of dipole, because pulling the fluid in from front and back. Its far flow field will be order 1/r square, and you can model this as a 2 Stokes-Slet system which comprises the force dipole. Experimentally, the flow field of this organism in an unconfined geometry has been measured, and you can see that it's actually a lot more complicated than simplistic picture. You have these two vortices that are closer to the cell body, and then you have two more circulating flows far away, and then you have a stagnation point in between, which is quite some distance from the cell body. And so to actually get all these features, you have to invoke what is called a 3 Stokes-Slet model, and that's mainly accounting for the fact that the cell body is, is kind of extended. It has these two appendages that are as long as the cell body itself, and so the force is sort of spread over, and you have two Stokes-Slets corresponding to each flagella, and then one Stokes-Slet corresponding to the cell body drag. And this kind of 3 Stokes-Slet picture lets you explain the unconfined experimental flow field. So that's sort of the background knowledge of Chlamy's flow fields. So we've been looking at these organisms in sort of these idealized laboratory conditions, unconfined flow and so on. But in reality, this organism sees confinement all the time. In fact, its natural habitat is soil, rocks and pools, where the organism is forced to navigate tight spaces. And in lab experiments also, you're looking at them in slide cover slip geometry or microfluidic channels. And so, and more recently there are these experiments where you see that the lipid synthesis in squished clammy cells is upregulated as you squish them more and more. And so, so it's not really a very theoretically motivated question as to what happens to motility and flow field under strong confinement. It's actually a question that's more motivated by its natural habitat, by its technological implications. And so, what is the theoretical understanding of flow fields under confinement? So, Julia and her group has done extensive calculations for flow fields of Puller type of microswimmers under confinement, and she showed that the far field would be that of a 2D source dipole oriented along the swimming direction. So this was all good, and this is applicable when the stresses in the system are purely hydrodynamic in origin. So now we started to look at this problem experimentally, and we measured the motility and flow field of CR in both weak and strong confinements, and I'll define what that is. So by weak confinement, what I mean is that we are looking at the cell which is which is put in a chamber which is 30 microns in diameter, or 3 cell body diameters, and here the flagella are essentially overcoming only the cell drag. When you confine the cell to a spacing which is comparable to the cell diameter, about 10 microns, then the flagella have to overcome the friction from the walls as well. So the free body diagram now changes, and let's see experimentally what is the repercussion of this change in the force diagram. So we'll define the degree of confinement as the ratio of cell diameter to the chamber height. So the chamber is completely unconfined in the xy dimension, and the only confinement is in one z direction. So here is the video of a cell moving in a 30 micron chamber, and you can see the tracer particles move a little bit. When you confine the system extremely tightly, you see that the cell is moving slowly, and you can see the vortices or you can see the flow field much more apparently. So and then so this sort of behavior in the extremely confined case has a little bit of detail, which is that you can have cells which are showing this synchronous movement of flagella, and then you can have cells which are showing whose flagella are behaving much more erratically. So the ones where the flagella are behaving erratically are called wobblers, and the ones where the flagella are moving much more nicely are called synchronous. So what is the difference between wobbler and synchronous is mainly the cell size. So here I am plotting a histogram of probability versus cell diameter for the two types of motion, and you can see that wobblers tend to be smaller compared to the synchronous cells. So our chamber is still 10 micron, but we have some heterogeneity in the cell size, and that heterogeneity is leading to heterogeneity in the flagellar motion. So we'll completely ignore these wobblers because they have irregular beat patterns, so it's difficult to actually measure any beat averaged flow field. Largely what's happening is that the flagella are undergoing contact interactions with the walls, and that's leading to, yes, you have a question?
Yeah. I don't know if it is just illusion or
Yes.
something. So if you plot the center of mass as a function of time, does it go back a little bit also?
Yes.
Oh, okay.
So it's in Clemidomon, it's very well known that it shows a back and forth motion, and so in the power stroke it will go forward. Power stroke, so you can divide the waveform into power versus recovery.
Okay.
And so while the flagella goes back, the cell would move forward, and then the other way move backward, and the forward backward motion is not symmetric. That's why the cell is moving.
Yes.
And so that's leading to back and forth. Yeah.
Thanks.
The background is just water, is it?
Yes, it's water with tracer particles, 200 nanometer tracer particles. And we had functionalized these tracer particles with polymers to prevent adhesion to cell body and flagella. Yeah. So, yes, Eric.
Is the beat frequency the same for the different levels of confinement?
Yes. Mainly because the flagella, for the synchronous cell as well as for the weakly confined cell, the flagella don't see the boundary. It's still 5 microns away. Yeah.
Prerna, are you always looking at one
cell?
One cell, or are they interacting with each other?
So these were very dilute suspensions. So we were looking at one cell, but we have looked at multiple cells also. And to some limit, you can do superposition and get the, if you're just looking at two swimmers, you can do superposition and get the flow field.
If it's dilute.
Yeah. So, so here I'm showing the trajectories of these 3 different cell behaviors. All the trajectories are, their starting point is mapped to 0, 0. And so you can see in the weakly confined case, you just have cells moving in random directions. In case of a wobbler, it becomes erratic. And in case of extremely confined synchronous swimmer, you get these circular trajectories. And so if you zoom in, you see the back and forth motion that Shayantan talked about. And then in extreme confinement, this back and forth becomes more loopy. And so you can characterize this by measuring the tortuosity of the trajectory. And so we plot the cell speed as a function of degree of confinement. The cell speed goes down dramatically as you increase the confinement, and the tortuosity goes up as you increase the confinement. So that's sort of the coarse-grained analysis of what is happening to the motility of these cells. Yeah, so essentially under weakly confined case, it's as good as bulk, about 120 microns per second of speed, which goes all down to all the way 4 microns per second in extreme confinement.
So how are you
Yes.
defining tortuosity?
So what's the definition of tortuosity? So we break this trajectory into certain time, 10 beat cycles, etc., and then compute the arc length versus the net displacement. Yeah, I mean it's a terminology borrowed from porous media. So
Okay.
yeah.
Thanks.
Right. So this is the unconfined flow field. This is the weakly confined, which resembles the unconfined flow field, but you can notice that the stagnation point has moved a little bit closer to the cell body. These black arrows denote the direction in which the cell is moving. And then in the strongly confined case, the stagnation point is extremely close, and this 4-lobe field has now turned into 2 lobes field. And what you also notice is that here the vortices were going anti-clockwise, and here the vortices are going clockwise. So there is an inversion in the sign of the vortices. So in fact, Sriram was crucial in this observation, because we had measured this and promptly displayed it on our computer screen. And he walked in and he said, the sign doesn't look right. So that led us down on a detailed path to look at why the sign is, the opposite way. So, right. So, also I wanted to mention that a quick way to understand what is happening between these two flow fields is that essentially these vortices have moved much closer to the cell body, and these vortices are pointed clockwise. So that's what is going on. Theoretically, you can account for the confinement by going back to simple-minded 3-Stokes-Slit model, and you'll get this 4-loop field, which does not compare well with the experiment. Or you could use a 2D source dipole with the Leron-Mochon approximation, and you will get 2 lobes, but they are of the wrong handedness. And so, so I've already told you the origin of flow inversion, and I'll remind you again. So essentially, as I said, in unconfined flow, the flagellar forces are overcoming the hydrodynamic drag. In the strong confinement, the flagellar forces are only overcoming largely the contact friction from the walls. So if we are now looking at the flow field, we should be thinking of Stokes-Slit corresponding only to the flagella, because this hydrodynamic drag is very small. And so this is what is causing the flow inversion, and all you need is a pair of like sign force densities on flagellar positions. So let's do that. So essentially what we are doing is we are taking a 3D Stokes equation and putting in, so now that we have extremely confined our cells, we can limit ourselves to the first Fourier mode of the velocity and get a quasi 2D approximation to Stokes equation. We use a Gaussian forcing because the flagella is an extended object, and therefore to account for that we have Gaussian forcing. Even if you, and Gaussian forcing is what lets us also get a good match to experimental data. So you can solve this theoretically and invert it to get the theoretical flow field. So here is the experimental flow field, and then here is the theoretical flow field with only two Stokeslets corresponding to the flagellar positions. The comparison is quantitative, so I am plotting the normalized flow velocity as a function of polar angle along the cell body, around the cell body, for representative radial distances, and we get a good match. So essentially, coupling between the motility and strong confinement alters the force generators. So that's what the previous theoretical studies missed out, mainly because they were not accounting for this coupling. And so this reduces a force dipole swimmer to an effective force monopole one.
Prerna?
Yes, yes.
So when you said Gaussian, you meant Gaussian in the distance, but what about the direction?
Not Gaussian in the distance, but in the strength. Yeah, okay. Yeah, so radially averaged, yeah.
But what is the direction of the forcing?
Here,
swimming
right?
direction.
It's no, it's the flagellar motion direction, right? So the two Stokeslets are Gaussian-like. These are pairs.
So there are two Gaussians.
Two Gaussians, yeah, yeah.
Can you actually go back to the equation and say that for people like me who are not
Sure. I'm sorry.
not
Yeah.
getting what's being put into the equation?
So here is my standard Stokes equation with the continuity equation, and then I'm using some kind of parabolic flow profile and substituting it here to convert a 3D one into a 2D XY equation. And then instead of having point forces, I mean, if you were doing Oseen tensor, then you would just put a point force. Instead of putting a point force, we are putting in Gaussian forces at the two flagellar positions. And then you solve it in the Fourier space and invert it to get the space in, I mean, real space flow field. Right. So all this is good. I have shown you how the motility changes, how the flow field changes, but is there any ecological consequence of all of this for the cell? And so we looked at what happens to the velocity correlation function of the flow field and found that there is a slight increase in the decay of the velocity correlation as you increase the confinement. And why we are really looking at that is because we are interested in how the fluid around it is stirring, and and that has consequences for how the cell gets nutrients essentially. And so so here I am plotting the maximum tracer displacement over 10 beat cycles for the two cases, the weak confinement and the strong confinement. So what you can see is that for the same amount of time, in the weak confinement, the cell will actually cross your field of view, and there will be little bit of motion along the cell trajectory. Whereas in strong confinement, the cell is going in a circle, and there is a much larger region of influence and much stronger tracer displacements. So larger advective displacement of traces and larger radius of influence as you increase the confinement. And you can plot the mean square displacement of the tracers as a function of time, and you find that in a stronger confinement the exponent is slightly higher. But more than the exponent, I think this picture is more reflective of, yes, Eric?
Yeah, so this is super interesting. I was wondering, does the stroke of the organism change in going to get you from the dipole to the monopole, or is it just that it's stuck, the friction from the wall?
It's just the friction from the wall.
It's just the friction from the wall.
Yeah.
And so, yeah, this is really neat, because if normally you would expect that when things are confined, all the correlation functions would be more short range.
Right.
And here
It's completely opposite of your expectations in that
And it's
sense,
all
yeah.
about the friction, I guess, is the key.
Yeah.
Thank you.
Thank you. Yeah, one of our referees asked what happens in between. How do you go from this kind of behavior to that kind of behavior? And experimentally we can't do it, and I suspect if even if we attempt, it's going to be like a first order transition, so we may not really.
Why do you expect
Okay, that's just intuition. Yeah, I don't have a good reason to say any other way.
You don't have spacers of a continuous set of values or sizes that you could tweak.
Okay, yes, I mean, that's not the ultimate constraint. See, one could make a PDMS chamber and pressurize it and continuously change the diameter. There was only so much we could do, I think. That's what happened, but yeah. Yes. Gaussian can fit it. I mean, Gaussian will, that was not the statement. Yeah, even for weaker confinement, you don't really need this kind of picture, because then you would put 3 Stokes slit and get it. I mean, all of this was important when it is when you're thinking about friction from the walls and yeah.
So I'm actually still missing it.
Yes.
What did the friction from the wall do? So what was the what would be the deconfined equations? And so did you just throw away the hydrodynamic forces? Is that what happened? I don't
I think the picture is okay. So what's changing is how many Stokes slits we are placing.
And why,
So
why does friction change that?
mainly because the hydrodynamic drag, the cell is moving much more slowly under.
So I can neglect the body.
Yes, you neglect the drag. Yeah.
So there's no viscous drag.
Yes. I mean, there's viscous drag experienced by the flagella. There is no viscous drag experienced by the cell body, because the cell body is moving slowly. Yeah. Yeah, the author is here. The theoretical collaborator is here. No, I think we settled it.
If you want to go between these two, then you sort of need to know what the relative.
Yeah, the point is here really it's moving so slowly, and whatever it's trying to push itself forward is balanced by the friction of the wall.
Yeah.
So it's as though it's just sort of sitting there and waving its hands.
Yeah.
And that's set by the
Yeah.
dimension.
Yeah.
So that her 1/h squared times the viscosity is not the wall friction.
Yeah.
It's just the viscous friction in the confined geometry. The wall friction has gone into cancelling the velocity. And you can pretend it's more or less completely
It's a tangential force that cancels out.
Yeah.
Can you just change the viscosity of the medium?
Yeah, we can do it. We didn't do it. Yeah, that's. But basically, because the speed is already down to like 96% lower than the bulk speed, if we put more viscous medium, it will basically stop. Okay, I mean, then it stops. Then it's like a stationary cell held on a pipette, and you have 2 flagella beating. Yeah. So we didn't pursue that. One could ask a question of, instead of having this hard glass plates, if we have soft polymers, I mean soft elastomer kind of objects, then it maybe the deformability of the substrate will do something funny.
Or if you want to change it continuously, then you put it in something like
Wedge kind of geometry.
variable thickness geometry.
Yeah.
So you get stuck.
Right. Yeah, there are many ways you can design experiments to explore a range of heights. Yeah. Yes.
I just wanted to comment
Yes,
on
go
that because I think if you put a viscous solution, the effect on the flagella will also be significant.
Yeah, that too.
Because here what we are seeing that an unconfined swimmer, the flagellar beat, and a confined swimmer, the flagellar beat is essentially same.
Yeah.
Because they are just 200 nanometer filaments, and they are not seeing the walls. It's 5 micron. But if we put a viscous fluid, it may alter the flagellar waveform.
Yeah, so the beat frequency is inversely proportional to viscosity of the suspension. That is well known. So that will change the, yeah. Thanks for the comment. Okay, so we did that. So far what I've shown you is that you can there was inversion of lateral flow vorticity when you squish these swimmers to their cell body diameter range, and you get enhanced fluid mixing despite higher friction. And so that has implications for the organism, because so usually the idea is that it is a free swimming organism and it will phototax or chemotax and go to regions of better nutrient availability. But in case it is in a very tight geometry of porous rocks and soils, then the flagellar beating helps it stir the fluid and get nutrients to it. Right, so that takes me to the next short part of my talk, which is taxis. And so these cells, the algae cells run towards light. Taxis is something which is very common in microorganisms. They would move in response to gradients of physical chemical stimuli. So sperms, for example, will run towards egg, and it empowers the organism to reach favorable conditions for thriving. And so biologists have traditionally focused on measuring taxis of single isolated cells. But we know that there are qualitative differences between the way a cell would swim in isolation versus the way it would swim as part of a densely populated suspension. So then one can ask, what happens to taxis at the collective population level? So this is the work of my other student Sujit, and so I'll give you a little bit of background to this. So you can do these experiments where you look at these cells in pitch dark. By pitch dark, what I mean is I'm imaging cells in a light where it is not responsive to phototactic behavior, and you would see that the cells are going in random directions, and it's like if you measure it quantitatively, it's a persistent random walk. In presence of light, they move towards the light source. If you look at this video carefully, you will see some of the cells are going in the wrong direction. And the question is that what happens to this fraction of wrong moving cells as you change light intensity and as you change cell concentration? So that is the basic question. But before that, we need to understand how does it detect light, how does it move towards light. So it has a primitive version of eye called an eye spot, and it has a bunch of photoreceptor proteins that absorb light, cause downstream changes in the flagellar motility to reorient the cell towards light. So there is this terminology of flagella being called trans and cis. So the flagella away from the eye spot is called trans and closer to the eye spot is called cis. So how does the cell scan its surroundings for light? It combines two features. One is that there is a slight asymmetry in the beating of the two flagella. They are basically phase slips from time to time. So you'll have synchronous beating and then little bit of a phase slip. And then the cell is also rotating about its body axis. So essentially you are combining yawing with rolling. So if you combine these two things, you will get a helical trajectory. And so essentially it's moving on a helical trajectory and rotating itself, and so it's scanning its 3D surroundings for light. You can visualize the eye spot using polarization microscopy. So its eye is made up of birefringent fibers, and so you can see this helical motion or this rolling of the cell body beautifully. And so so you can measure this rotation frequency by doing this kind of imaging. So what happens in presence of light stimulus? What happens is that in presence of light stimulus, the flagella that is closer to the eye spot will have its beating will be inhibited, and the flagella that is away from the eye spot, its beating will be upregulated, and so the cell will turn continuously. So cell will turn towards the light, and then when the eye spot is in the dark area, when it's facing away from the light spot, it's the other way around. So the cell keeps turning towards light, essentially. And so you can really visualize this at a single cell level by holding the pipette on by holding the cell on a pipette. So these are some classic experiments, because in those days high speed microscopy was not there. So they measured it stroboscopically, and now with more modern tools you can see that in absence of light the two flagella are beating similarly. In presence of light, the flagella closer to the light source is moving much more slow, its amplitude of beating is smaller compared to the one that's away. So that lets the cell turn towards light. So here, so this single cell behavior is known, but free swimming behavior is missing, and phenomena at the population level is unknown. So there are these some old experiments where what they did is they have a stimulus light that's that's falling on a tube containing cells, and then you have another light which is measuring the optical density of the suspension. So what these people found is that the rate of change of optical density is a function of cell concentration. So in that sense, it was known that phototaxis of Chlamydomonas is collective in nature because there is cell concentration dependence. The way a cell would phototax in isolation is different from the way a cell would phototax in presence of other cells. And more recently, there was high-speed videography to plot these trajectory vectors and get the same information. But what remains unknown is why collective phototaxis is taking place, and that's where we come in. So, so far what I've told you is that phototaxis is concentration dependent, the origin remains unknown, and the kinetics of phototaxis is also unknown at the population level. And with modern day tools, we can answer many of these questions. So our experimental setup was very simple. We have a chamber filled with cells. We are shining blue laser light at one end of the chamber, and we are visualizing cells far away from this light. So these cells are actually extremely sensitive to light, and therefore even such a weak gradient was good enough for them to run towards light. And so here again the cells are moving in the dark. One can measure the mean squared angular displacement versus time, and in the long time limit it's a linear behavior. So you can get a rotational diffusion coefficient. So it's important to know what is the rotational diffusion coefficient, because that tells you how the cells are going to deorient with respect, in presence of other cells. And what we found is that the rotational diffusion coefficient is independent of cell concentration. So over a large range of cell concentration. So this detail is important. And so now let's look at in presence of light. This is the movie I had shown you earlier. And now you can plot the cell trajectories. So again, the trajectories are color coded for clarity arbitrarily. All the trajectories are set to 0, 0, and you can see that there is a bunch. So here the light direction is in this direction, and so most of the trajectories are along the light direction. So you can ask what fraction of cell trajectories are within 20 degrees of the light stimulus, and plot that as a function of light intensities, and as expectedly with increasing light intensity that fraction goes up. One can also plot a probability density as a function of polar angle and see that in the dark it's a flat distribution which then builds up along the light direction as you increase the light intensity. So far it's all standard expected. Now let's fix a light intensity and change the concentration. The light is in this direction. Okay. So initially most of the cells are going in the right direction, and there is some confusion, and that confusion goes away as you increase the concentration further and further. So in other words, if you plot the probability density, you will see that the peak will go down and then go back up. So there is a re-entrant behavior of the probability density. And I can plot the phototactic efficiency as well and see the same behavior. What we see is that at lower light intensity, this re-entrant behavior is much stronger compared to higher light intensity. we don't understand why the efficiency should go down initially. What I'm going to show you is a mechanism for how the efficiency could increase as you increase the cell concentration. And later on we did this experiment for large number of light intensities and cell concentrations and found similar reentrant behavior. So the modeling here was done by Professor Aparna Bhaskaran from Brandeis, and she had this model where you think of the cells as active Brownian particle and the light field as a torque that aligns the cell along the light direction. And so this torque is like an external potential of the angle dependent potential of the form gamma cos theta. And then you write down the equation of motion of single cell. So this is a translational equation of motion and this is the angular equation of motion. And then you can write down a Fokker-Planck equation to look at the collection of cells or the probability density, whose solution is a von Mises distribution. So it effectively looks like a Gaussian distribution, but in the form it is similar. Sorry, in the form is different. And so this is our fitting function, and the only fitting parameter here is the ratio of the torque strength to the rotational diffusion. Since I had shown you that the rotational diffusion is independent of cell concentration for us, so the only thing that's changing is the strength of the torque. And so we can get very nice fits to our experimental data and extract some kind of effective torque. And that torque has a very similar variation as the experimentally measured phototactic efficiency. So, so this simple-minded model captures our experimental data very well, with the caveat that you have a density dependent torque, the cell density dependent torque. So then, so mainly the question is what is going on? Why are the cells showing better phototactic efficiency as you go to denser and denser suspensions? And so we initially thought, okay, maybe there's some kind of quorum sensing that cells are moving and leaving a trail of chemicals behind which other cells can follow and reach the light source. And then we tried many experiments by exchanging media, etc., and nothing seemed to make much of a difference. There is a physical phenomenon that also happens, is that the cells slow down as the cell concentration increases. So there is a density dependent motility. So I am plotting the mean speed as a function of cell concentration, and I see that it goes down. And in pitch dark also that is the case. So both in presence of light and absence of light, the speed goes down as you increase the cell concentration. So it's more like a crowding effect. And then how does this crowding help me in detecting light better? And that's mainly because if the linear speed comes down, then so does the rotational speed of the cell. And if you're rotating slowly, you're collecting more number of photons per half a rotation, and therefore you have better chances of detecting the light direction accurately. So you are integrating the signal a lot more and making less number of mistakes. And so this is again the same video where we follow the eye spot, the cell body rotation, and we measure the rotation frequency as a cell concentration and found that it is also going down. So, so if this mechanism has any value, I should be able to independently validate this hypothesis. So instead of slowing down the cells by increasing concentration, I will slow down the cells by adding a polymer like a methyl cellulose, as someone was suggesting. So here we did change the viscosity, and we found that, yeah, sure enough, you will decrease the speed of the cells as you add more methyl cellulose. And so we fixed the cell concentration, changed the viscosity of the background medium, and plotted the phototactic efficiency as a function of mean speed. So the green data points are the data points that we obtained by changing the cell concentration, our original observations. And then the pink data points are the one where we have fixed the cell concentration and we are just changing the methyl cellulose. So these two overlapping curves tell you that indeed this mechanism of enhanced efficiency is mainly coming from the changes in the speed of the cell. So this is a purely physical mechanism of enhanced phototaxis. And so now I'll conclude that I've shown you how confinement changes the flow fields. I've shown you how phototaxis of Chlamydomonas is collective in nature and it's related to changes in the speed of the organism. So with this I will stop. If there are any questions, I'll take it, otherwise we go to coffee.
Very nice. I have two questions.
Yes.
I guess one is a sort of a suggestion. I don't know if you've
Yes.
tried it, but if I put together the two halves of your talk,
Yes,
and I have a whole bunch of clammies squeezed together,
yes.
can you make a nice microfluidic pump that's optically driven?
People have done that.
Oh, okay, okay.
It's a very, yeah, I mean, it's a popular theme in this biofluid
I
mechanics
see.
community. And
Okay,
life in moving fluids, yeah.
Cool. Okay, and then the more real question is, you showed that the rotational diffusion
Yes, yes.
was independent
Yes,
of the concentration.
yes, yes.
And that the velocity goes down with concentration,
Yes.
which is the same as increasing the viscosity.
Right.
But I would have thought that if you increase the viscosity, that would have decreased the rotational diffusion. So somehow it seems
yes,
like concentration and viscosity
yes.
are
So if-
not quite the same, or?
So I showed data where when you increase the cell concentration, the rotational frequency also goes down.
Oh, it does.
Yeah,
Okay.
yeah,
Oh,
so-
I thought it didn't.
Yeah,
I thought it didn't.
yeah.
Okay.
Rotational frequency. I mean,
The frequency or the diffusion coefficient?
frequency, the cell body rotation frequency. So
Okay.
it's all related to how the cell is detecting the light.
But the long-term angular diffusion coefficient
Is the same, yeah.
is independent
Yeah,
of
it's
that. So
independent.
that's, I shouldn't, somehow that doesn't care about the viscosity.
Yes, you are right. I mean, we haven't measured the rotational diffusion coefficient by changing the viscosity of the medium. We have
Yes.
only changed it by changing the cell concentration and found no dependence. I suspect that okay, I think you have a point. I think we did these measurements for methyl cellulose. Yes, the DR was independent in methyl cellulose as well. Yeah, yeah, yeah.
Cool.
Yes.
I'm curious about the uniformity. So are they all identical, or are there some young or some old and tired?
Yeah, so these are what we call synchronized cells. So where they all divide at the same time, and then we always pick up our cultures at well-defined time points. So these things are light-dark adapted. So they would see this day-night cycle that we artificially give them. And at the start of the light period, they will divide exactly all at the same time, and then we'll pick up 2 hours in the light cycle. And then all these phototaxis experiments, you again dark adapt for half an hour before you show them light. Yeah, I mean, these experiments are actually very challenging to, to get this kind of robust statistics. So yeah, there is, I mean, in any biological system, there will be heterogeneity, so there will be some differences. But as I've shown you, the heterogeneity has more of a physical basis here than just to say this is, genetically diverse population where some fraction is going in the wrong direction. Yeah.
What's the average life of
These cells divide every 12 hours, and they will keep on going as long as you give the right food. Original also remains. Meaning?
once it divides, okay.
Yeah, it'll just, they will mate. And
Otherwise immortal, is it?
Yeah, it looks like.
yeah, if you give them food and light all the time, that's how we've been maintaining these. So these cells you can't freeze, so you have to maintain live cultures. Yeah. So for 10 years we have kept them alive. So I'm sure we can maintain them.
Yeah. Do I remember correctly that clammy produces more oxygen than, well, yes,
Yeah.
and consume carbon dioxide?
Yeah, they are
So
responsible for 50% of
So
oxygen production.
do some people work on modifying genetically clammy so that it
Yes.
even
Yes.
consumes more
storage
CO2?
and, yeah.
The people do that as well.
Yeah, in the fields of biotechnology, that's what they're trying to do.
Okay. So you're going to be rich.
I hope so.
Yeah, so So I was wondering about the spread of the trajectories.
Yes,
like
yes.
you have a light
Right,
source and
right.
how tightly they're
Right.
swimming. So you mentioned that it depends on the flux of photon they're
Yes, yes.
detecting. So I was wondering that to test that, suppose you don't change viscosity or anything, your rotational diffusion is same.
Yes.
Now, if you increase the light intensity, so
yes.
they are collecting more photons.
Yes,
so
yes.
the width of the distribution narrows
Yes,
down.
yes, it does, yeah. I mean, not the width, the peak of the probability density will build up. So I was plotting the angular probability density.
Yes.
So the width stays more or less the same, but the peak goes up because now more number of cells are going towards the light source. Yeah. Yeah. That's
Less
a sanity
spread in
check.
the
Yeah.
angle.
Yeah. That's a sanity check, right? That with more signal, they should be able to do better.
Actually, I had a follow-up question on
Yes.
that. If you go to the probability distribution, the angular distribution,
Yes.
there were some of them which were going the wrong direction to
Oh,
light.
No. Actually, I didn't do a good job of showing where the light is. Is here or sorry, I'm not on the. Okay, let me quickly go and. This is as a function of cell concentration.
No. You had one much earlier where you had.
Much earlier. Those. So next.
Yeah. No, there. Go back.
Yes,
Yeah, there.
yes.
So the light source is at 3 by 2. Is that right?
I think light source is here. It's this angle. You should not take this. I mean, this angle is arbitrary, right?
That's
Depends
fine.
on.
But relatively, it's a 3 by 2.
Yeah.
But there's also a peak at pi/2. Is that right?
This is noise. Yeah. No, that's
noise.
not. Yeah.
Then what accounts for the things that are going in the wrong direction to light?
Yeah, but I mean, I wouldn't take this peak very seriously. If we would have taken more data, it would have got smeared out.
I mean, there's at least two different.
Red and yellow.
Red and green,
glowing,
yeah. Yeah, I mean, both of these have it. Blue doesn't have it, so that's why I wouldn't worry. Yeah, I mean, so what you can see is that the blue the base level has gone up, right? So, yeah.
Is there any saturation intensity, if you keep on increasing the
Yeah. There is in fact, if you give it too bright a light, it will become photophobic. It will run away because it will cause photo damage to the cell.
So it
So
knows
then
how to run away.
Yeah.
So
It
there's
can
some
do.
mechanism for that.
Yeah, it can do negative phototaxis. So I've shown you positive phototaxis, but it will do negative phototaxis if you give too bright a stimulus. Yeah. And there's very interesting phenomenology to what happens at the kinetics of the cell. Yeah. It will stop and then run away.
There's a question at the back.
These cells respond, they give positive phototaxis to blue light and negative phototaxis to green light. And so in that sense, there is wavelength dependence in the sign of the phototaxis. But I don't know how this results would translate if you do it on a different wavelength. And these are essentially photosynthetic, so they will respond to a broad range of wavelengths. And people have measured at these two because these are two nice laser lines people can do experiments with. Yeah.
Your motility data as a function of concentration
Yes.
had some slight monotonicity.
Yes.
Non-monotonicity.
Non-monotonicity,
Was
yes.
slightly non-monotone.
Yeah, so let me switch to this data. Yeah. Yes, you're right.
We can't see it though.
Yeah.
Yeah.
Here, yeah.
Yeah, so what's going on, do you know?
Okay. So I would rather look at blue and red data here. So below the critical concentration, there is a small flat region and then there is a decay. So this is in pitch dark. I look at the size of my error bars and I wouldn't take this, I wouldn't worry about a small peak here. I would still consider it as a flat and a fall off, gradual fall off, yeah.
Okay.
Yeah.
But there is a non-monotonicity in this gamma coefficient, right?
Yes.
And where does that come from?
Right. So as I said, maybe I'll go to that plot and then it'll be easier for the rest of the audience to. Yeah. And so So I only talked about this regime, above a critical concentration. Below the critical concentration, I don't know, but I actually don't know the mechanism, but most likely it's related to self-shading. That, you know, when you start increasing the concentration, initially they are just absorbing the photons, and so the other cells are not blocking it as some kind of shadow, and then
So the collective effects
haven't shown up, and yeah. And, I mean, if it were purely shadowing, it'll just keep decaying, right? But it doesn't, and yeah.
Other questions, comments? Okay, so let's thank Prerna for a very nice talk. So we break for coffee and come back at 4 in 35 minutes.
She's just thinking. Yes.
Okay, welcome back everyone after the coffee break. So it's a pleasure to have Irmgard Bischoff-Berger from MIT. So she'll tell us about pattern growth in flow-structured complex fluids.
All right, thank you so much. It's really an enormous pleasure and honor to be here. So I'm looking forward to a week of exciting science and to meet a lot of new people. And so I want to start this talk by asking a question that I'm not going to quite answer during my talk. Why is there so much structure, order, and texture to life and nature rather than a featureless expanse? And so wherever we look around us, and even within us, nature has created an abundance of patterns, from nanoscale DNA to majestic river networks. And so for us as scientists, this emergence of pattern provides a uniquely fun playground, but also some really deep and challenging questions. So pattern growth usually occurs in systems that are far from equilibrium, and the growth of patterns is typically nonlinear. And so far from equilibrium system and nonlinear growth are really two concepts in physics that we know least how to think about. In addition, in these pattern-forming systems, very small perturbations to the system can lead to a large-scale response. And so in my lab, we study pattern formation in complex fluids and soft materials, with the goal to develop a fundamental understanding of pattern growth in order to tune, control, and predict pattern growth in complex fluids. And we aim to exploit the control we can gain over the structure selection in order to design soft materials with tunable properties. And so I want to introduce the people who do all the work. This is my current lab, Ippoliti, Delace, Mario, Paul, Sami, and Ching. I'm really incredibly lucky to get the chance to work with them. And so what I want to tell you today is two stories, two ongoing stories of how flows which push a system out of equilibrium and might be thought to actually destabilize any texture can instead induce structure in complex fluids. And so I'm going to tell you about dense suspensions, and I'm going to tell you, given the place I'm at, about structure formation in liquid crystals. So dense suspensions are ubiquitous as construction materials, chocolate, geophysical materials, and the particles at high concentration suspended in a liquid. And so I hope you've all, at least once in your life, played with one-dense suspensions, cornstarch in water, which is the system we're going to use in this talk, where you probably know that as you apply a high stress to the material, you can fracture it, so it behaves like a solid. However, as you release that stress, the suspension can flow like a liquid. And so over the past 15 years or so, the rheologists, including Sayanta and here the Raman Institute, have taught us how to think about these complex material properties. So dense suspensions are shear thickening fluids whose viscosity increases with shear rate beyond a certain shear rate. This increase can either occur continuously with shear rate or discontinuously with shear rate, giving us discontinuous shear thickening, depending on the mass fraction of cornstarch in water. Now, there's another way to look at this data, which is to plot the viscosity as a function of the shear stress, where in continuous shear thickening, we have an increase with the viscosity with the shear stress with an exponent smaller than 1, and a linear increase of the viscosity with the applied stress for discontinuous shear thickening. Looking at this stress controlled also allows us to see or to enter and probe the shear jam state that occurs at high enough stresses where the material behaves like a solid. Now, this discontinuous shear thickening and shear jamming is a response of a flow-induced structuring in the material, where at low stresses, the contacts between the cornstarch particles are lubricated and the material can still flow. However, as we push the system to high enough stresses, the contact between the particles become frictional. A space-spanning contact network forms, which leads to the onset of discontinuous shear thickening and at the highest stress to shear jamming. Now, both the viscosity of the suspension, which we here take as a measure the minimum viscosity, and the shear rate at which the material starts to shear thicken change monotonically with the mass fraction of cornstarch, where the minimum viscosity increases and the critical shear rate denoting the onset of shear thickening decreases over order of magnitudes over a very small range in mass fraction of cornstarch. And so what I want to tell you about today is what the consequence of this complex rheological behavior is for air injection into a suspension.
Just a question.
Yeah.
Can you explain operationally, experimentally, what is meant by the double-valued viscosity versus shear rate in the left graph? The red dots on the leftmost, the leftmost, yeah.
So this is different mass fractions. So at low mass fraction, I get the discontinuous shear thickening and a fairly low viscosity.
Right.
At higher mass fraction, I transition to discontinuous shear thickening and the viscosity.
No, but how do you measure the double value, the double value viscosity?
So the way we run the experiment is we run it as a stress control experiment.
Okay.
And so we have stress ramps going up and down, and then we plot it as a function of the shear rate.
Okay.
Yes, thank you for asking the question. All right, and so in order to look at air invasion into these suspensions, we focus on the regime of high mass fractions between 57% and 60% of cornstarch in water, which is the regime where the suspension experiences discontinuous shear thickening and can shear jam. So in order to look at air invasion, we need to be able to look inside the opaque suspension. So we use X-ray radiography, where we have a X-ray source in front of a container that is filled with a bulk suspension of cornstarch in water, which is open to atmosphere at the top, and we have an X-ray receptor that measures the transmitted X-ray intensity. Now, the resulting X-ray image is going to be a projection along the thickness of our container. And in this visualization mode, the cornstarch in water will appear as black, and the air which absorbs X-rays less efficiently will appear as white. Now, we inject the air from the bottom of the container, where the air into the suspension, where the air comes from an air tank that is filled to a certain gauge pressure, PG. So for a high mass fraction and a certain gauge pressure, we see that the air invasion leads to a fracturing of the suspension. Now, to quantify this air invasion, we convert this transmitted X-ray intensity to air thickness by calibrating with cornstarch containers of different thicknesses and using Beer-Lambert's law. And so at this high mass fraction, we see that air invasion fractures the material. If we decrease the mass fraction slightly, we still get fracturing. If we go down to 57% of cornstarch in water, we see that the air invasion transitions to a bubble. This is for...
in which regime?
Yeah, I'm getting to that in just a second. So it's for a fixed gauge pressure in this case. And I'm going to tell you in a second how we should think about controlling the pressure in this system. Give me one slide. Yeah, we essentially open a valve and inject air that is in the tank at that specific pressure. I'm emphasizing that because we actually have to think a little bit what's happening here in terms of how these bubbles grow. And that's the first thing we want to do, is to look at the growth dynamics and at the motion of these bubbles. Yeah.
Is it density matched?
We do both.
Oh.
So most of the experiments I show you are not density matched. We are careful in preparing our suspension such that it's fresh, such that the suspension has time to settle. In order to make sure that settling isn't an issue, we have done control experiments that are density matched. Yeah.
Okay, thanks.
So just to understand that, your volume is constant and here that you open up the pressure, is the volume constant?
Yeah, so we have a constant pressure in our air tank. We open the valve, we let a first bubble appear, and then we stop the experiment. There's a lot of really cool stuff happening if we just let the pressure valve open. Then you get multiple bubbles, multiple fractures. The bubbles are going to select certain pathways that seem to have a memory of the previous bubble. But today, I'm only going to tell you what happens to the very first bubble, and then we close the valve again.
So you find out how, when the first bubble has ended by looking at some pressure and the pressure dropped. That's how you cut it?
Yeah, we see that we measure the pressure, we have a trigger, we close the valve after we know that the first bubble has fully formed and fully grown. I think there was another question.
How reproducible is it? Will you always get fractured?
Everything, yeah. Everything I'm emphasizing today is very reproducible. The exact shape of these fractures, for example, is not reproducible. And that's given by potentially some small variations in density of the suspension at the nozzle inlet. But, so the shape is not reproducible, but the fact that it either fractures or forms a smooth bubble, that is reproducible. Yeah.
Why do the fractures proceed in one direction rather than?
That is the part that is random. So the shape of the fracture is somewhat random. They generally, but not all of them, try to form a penny-shaped crack, just like in a solid elastic material. Now, different from a solid elastic material, where a penny-shaped crack is going to grow in the lateral plane, here the suspension, because part of it is still fluid-like, can actually expand and widen this fracture in the way you see here. Yeah. Oh, it's very, 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 very low. Yeah, 10 to the minus 5 or something. It's very low, yeah. Yeah, you see some, right? So if I go back to the, especially at the higher mass fractions, so the brighter features you see here are air bubbles that are initially trapped in the suspension. They're mostly trapped at the container walls. They don't seem to play much of a role for the things I want to emphasize today. All right, so back to the question about how do these bubbles grow and what is it really that we are controlling? So in order to probe this bubble growth dynamics, we sum the air thickness over our entire image to get a bubble volume. And this bubble volume increases linearly with time with a constant flow rate that is set by the gauge pressure of our tank, but that is independent of the mass fraction of cornstarch. Now, this took us a while to wrap our head around why we observe that the bubble volume increases linearly, why we have a constant flow rate that is set by pressure but not by mass fraction. The reason why that is, and that's why I was very careful with what I was saying before, is that what's actually happening here is that the pressure loss in the tubing between our air tank and our nozzle is larger than the pressure loss that happens at the bubble interface, which means that even though we control the pressure in our experiments, the experiments are effectively flow rate controlled. So we experimentally control pressure, but what the system does is essentially the pressure simply sets a specific flow rate in the material. Does that make sense? Excellent. So while the volume rate is independent of the mass fraction, the vertical motion and the growth of the bubble actually depends sensitively on the mass fraction. And we see that nicely by comparing the lowest mass fraction we observe with the highest one that we probe. But we see that at the lower mass fraction, the bubble detaches and rises in the suspension, whereas for the, which we nicely see in these contours, whereas for the 60% mass fraction, the bubble doesn't detach. The bottom of the bubble is essentially stagnant, and the bubble simply continues to grow without detaching. Now, to understand these differences in the vertical motion of the bubbles, we consider the vertical position of the center of the bubble as the bubble develops, and we see that this vertical position increases faster than the equivalent radius, assuming this is a spherical bubble, of our bubble, which tells us that we essentially have buoyancy acting on our negatively buoyant bubble, which makes the bubble rise, but we can express the buoyancy forces acting on our bubble as the density difference between the air and the cornstarch suspension, the gravitational constant, and the volume of our bubble. At the same time, we have viscous drag forces that slow down the motion, which depend on an effective viscosity of our suspension, on the bubble radius, and on the vertical velocity of the bubble. Balancing these two terms then gives us an expression for the vertical velocity of the bubble due to buoyancy, which indeed nicely fits our data. At the higher mass fraction, though, the fact that the bottom of the bubble doesn't rise tells us that buoyancy is here not acting. The bubble cannot grow downwards, so its center of mass still moves upwards as the bubble is growing. And we effectively have the viscous drag forces here that are way dominant over the buoyancy forces because of the high viscosity of the suspension in this state, which leads to a quasi-static growth that is independent of the flow rate that we control by setting the pressure. Now for a bubble, yeah?
So in that continuous shear thickening,
Yeah.
Is it fair to say that the viscosity is completely independent?
Yeah, and we'll get back to that. So it's absolutely not. But as we'll see, there's two very distinct regimes. And I'm going to tell you what they are and why, for now, I'm just telling you there's an effective viscosity. Yeah. All right, so for a bubble now that is simply growing without experiencing buoyancy, and that is growing with a roughly self-similar shape, we expect that the vertical position increases in proportion to the equivalent radius, which gives us a vertical velocity from pure growth that is given by a constant times the time derivative of the bubble radius. So this buoyancy-dominated regime and this growth-dominated regime are two asymptotes when we plot the rate of change of the vertical position over the bubble radius versus the ratio of the buoyancy velocity over the growth velocity, where we see that we have a growth-dominated regime for the high mass fraction, a buoyancy-dominated regime for the lowest mass fraction we probed, with a transition between the two growth regimes at a velocity ratio of 1. Now, in between these two extremes of purely growth-dominated or purely buoyancy-dominated regime, we can, for intermediate volume fractions or mass fractions of cornstarch, see a transition between growth-dominated and buoyancy-dominated during the growth of the bubble, which we see most clearly in these contour lines, where we first see a stagnation of the bottom of the bubble, but then the onset of a rise of the bubble due to buoyancy. Now, this transition occurs because the growth velocity decreases with the radius of the bubble, whereas the buoyancy velocity increases during the growth of the bubble, such that we transition from one regime to the other. So we get this transition from growth to buoyancy as either the volume of the bubble increases or as the mass fraction of the suspension decreases. And now I want to come back to your point of, is it fair to put just one effective viscosity? And it's fair, or roughly fair, in the buoyancy-dominated regime. And that is because we know that given that this is a buoyancy-dominated regime where the suspension is in a liquid state, and we know that these, in the liquid branch, these suspensions are very weakly shear thinning, we have an approximately constant viscosity there. It's absolutely not fair in this growth-dominated regime, but here, given that this is purely growth-dominated and doesn't have a viscosity dependence, we are kind of safe by just saying we are too viscous or too solid-like, such we are only growth-dominated. All right, so the mass fraction changes the regime from buoyancy-dominated to growth-dominated, but at the same time, and related to this change we just saw, we always have this maybe more dramatic change in the shape of these fractures, where we transition from smooth bubbles to fractured bubbles with an increase in the cornstarch mass fraction. Now, in order to quantify these fracture characteristics, we consider a fixed volume of the bubble, and we calculate the bubble thickness gradient intensity, where h here is the air thickness. Now these images of gradient thickness intensity, or thickness gradient intensity, show these sharp dark lines for the higher mass fractions. We associate these with local sliding cracks at the bubble interface. So we take this as a measure of the fracture characteristics of our bubbles, which are absent in these smooth bubbles at lower mass fraction. Now, given that this gradient intensity has a high value as we approach the bubble edge, even for the smooth bubbles, given that the air height goes towards 0 there, we quantify the gradient intensity thickness inside a bubble boundary that is the bubble outline shrunk by 5%. And we calculate the histogram of the gradient intensity inside this bubble boundary. But in particular, we are interested in the high gradient content of this histogram, which is the signatures, or which represents the signatures of the local fracturing. So this high gradient content decreases with decreasing mass fraction of cornstarch. We integrate the signal under these curves in order to get a measure for these fracture characteristics, which increases with increasing mass fraction of cornstarch, denoting that transition from smooth bubbles to more and more fractured bubbles. Now, yeah. Yeah. We would see quantitatively the exact same thing, qualitatively the exact same thing, but the fracture we would see the fracture from a different plane. Yeah.
Just again, following up and
Yeah.
clarifying. So you're changing the volume fraction, but is the flow rate also changing?
No. So as I showed in the, so for these sets of experiments, the pressure is fixed. The change in pressure also changes slightly this fracture metric. The higher we go in pressure, the more of these sliding cracks we're getting. Yes. So this tells the same story, right? By either increasing the pressure or increasing the mass fraction, the material enters a more and more, or becomes easier to locally fracture.
Yeah, I was just trying to see where in the phase diagram of a shear thickness.
We're up there. We're way up there. If you remember the rheology I showed in the very beginning, we're at the highest range of concentration one can experimentally prepare in a homogeneous way. And we believe, and I'm getting to that, that this is a signature of the shear jamming. Yeah. Yeah. So X-ray radiography is wonderful, but it's very slow. This is also why we can't go to lower mass fraction than 57%, because the fastest we can image is 4 frames per second.
4 frames per second is still fast enough for them not to anneal?
Not to?
Anneal, cracks of two just...
Yeah, so because they essentially remain, so during the entire growth, they remain attached to the nozzle, so they are constantly under the pressure that we are applying, until eventually, if I would let these grow to an even longer point, they will breach at the top interface and open up to atmosphere, and then the whole fracture collapses, and I'm getting a new one that starts to build up. Yeah.
Sorry, I'm not quite getting the link between fracture and the grad H. So are you talking about where the number of cornstarch molecules sort of goes down to 0 in the air and the interface looks like this? Is that how I should be thinking about it?
I'm essentially asking if I have any kind of discontinuous changes in the thickness of the bubble. Yeah. And that's what this gradient intensity captures. Yeah. And I associate these very rapid changes in the thickness with a fracture plane at the bubble cornstarch interface.
So it's going from bubble to solid? Is that the interface?
So at the interface between the air bubble and the cornstarch suspension surrounding it, I have a local fracture that gives me that signature that I'm picking up in this gradient intensity.
Okay, thank you.
you are controlling the pressure, so you are controlling the overall shear rate, we can say. But around the fracture, where actually the thickness arises, if we want to calculate, okay, let's calculate the shear rate around that, then we can say what sets the fracture exactly.
Yes, I'm getting there. This is wonderful. I'm taking this as a segue. No, you can still ask a question, but this is exactly what I'm going to tell you next.
Yeah, so I was wondering that you showed from 57 to 58 onwards, like there is a defined behavior, right? One is independent of mass fraction, another one is. So there is a discontinuous behavior there.
Yeah,
but this method, like it is a continuous.
Yeah, I'm getting there. All right, you guys know my talk, or it feels like you do. All right. So first of all, the fact that we see these fracture characteristics, right, seems to imply or to suggest that the suspension actually has entered a truly solid-like shear jam state. Now, this doesn't seem unreasonable because, again, we are in our experiments at these very high mass fractions of cornstarch where the material does experience discontinuous shear thickening beyond a certain critical shear rate and can enter a shear jam state at high enough stress. Now, coming to your question, what's essentially happening during the growth of the bubble is that I induce, say, a shear rate at the bubble, in the suspension at the bubble interface. And so if I consider a fluid element at the bubble interface, I see that as the bubble grows, I have a radial shear rate. The radial one is here the highest one compared to the other ones, so the material does experience a finite shear rate. And so given that all of our bubbles now experience a fairly high shear rate and can enter shear jam state, maybe the question to ask is more, why do not all these bubbles just fracture? And so the reason for that is that the shear rate at the bubble interface is highest at the very early times of bubble growth, but then decreases as the bubble continues to grow, which actually allows for something maybe some of you have picked up in the first videos I showed, a fairly cool phenomenon of actually seeing bubble relaxation during the growth of the bubbles. And so for that, we want to have a slightly closer look at what's happening in the 57% and the 58% mass fractions. But we see that indeed at early times, even these lower mass fractions fracture, but then relax into smooth bubbles. Likewise, for the 58, we see an initial fracture and then a relaxation. Now, we can again follow this relaxation as a function of time by looking at the histogram of the gradient intensity, where we see that the high gradient content monotonically decreases with time, indicating that transition towards a smooth fracture, where we can again get our fracture metric, where we see that indeed it decreases with time, denoting that bubble relaxation. We see a similar relaxation for 58% mass fraction. Now comparing the shear rate at the bubble interface that decreases as a function of time with the shear rate measured in a rheometer that denotes the onset of discontinuous shear thickening, which is for the 57%, this red line here, we see that the shear rate quickly decreases below that critical value, denoting the onset of discontinuous shear thickening. And indeed, for both the 57 and the 58% mass fraction, we see that the time at which the shear rate falls below the onset of discontinuous shear thickening corresponds to the time at which the bubble interface starts to relax. And that maybe answers a bit your question of the transition during the bubble growth. And so, to me, this is exciting because I believe it shows really a structural signature of this shear jam state in the form of these fractures, which occur as we have air injection into bulk three-dimensional suspension, where we see that we get a transition from solid-like fractures to liquid-like bubbles, as the shear rate at the bubble interface decreases below the onset of the shear rate needed for discontinuous shear thickening, and where this transition between fracturing and relaxation to smooth bubble is reflected as well in the bubble growth dynamics in these two distinct growth regimes, either being buoyancy dominated or growth dominated. So this would be a good time to ask questions, because in just a second I'm going to change a little bit the focus.
So I'm curious about, so if this was an elastic medium,
yeah,
and you had a crack that looked like that,
yeah,
the pressure would concentrate stresses at the crack tip. But what's interesting here is the crack tip should also have the very highest shear rate, and also should be like the toughest part.
Yes.
And I'm curious how
Yes,
that affects,
and that's
if you thought
exactly-
about how it affects the propagation.
Yeah, that's exactly why if I go back, or at the very end I showed this movie again, and if you look at it carefully, you see that actually in the very beginning you have something that looks like an elastic crack,
Yes.
which just propagates in the lateral
Yes.
direction.
Yes.
But then also because the fracture starts to feel so the shear rate slows down, the
Yes.
fracture starts to feel the walls.
Okay.
It essentially manages to widen, which an elastic fracture can't do. But I
see.
believe the very early part is very similar
Just
to
like
a true
normal
elastic
fracture.
Yeah, yeah, yeah.
Okay.
We can't look at it more carefully because everything happens more rapidly than we can actually visualize properly. So it becomes a bit tricky to really look at.
Do you have this X-ray thing in your own lab, or are you going to
No,
the doctor's office or something?
We use the one at Schlumberger, so
Nice.
we collaborate with Schlumberger, and they have one. Yeah.
Nice.
But they don't come faster than 4 frames per second.
Yeah, I was just wondering, what's the structure of the medium at the interface in the two cases? Is it like, when you have crystal in equilibrium with vapor, it can be faceted or what you call smooth, which is what the crystal people call rough, meaning that is you have a roughening transition from faceted to roughening.
Yeah, depends on which
And then,
scale you're looking at.
Right. But I mean, so what is the what does it look like if you at the interface?
I mean,
In the two
I can
cases.
tell you what I think it looks like, but I
don't
can't
really know.
visualize it,
I
right? But I think that it's really the transition that the rheologists also tell you about between essentially lubricated contacts, that's the smooth interface
Right.
between air and the suspension, and something that is in frictional contact and essentially can fracture. But
I see.
we can't, we unfortunately can't put a camera in there and look properly.
Thanks.
Hi. All right. So at the beginning of the talk, and you talked about the transition between the bubble rising and the bubble fracturing. I expected the word yield stress to show up at some point, and then it never did, and you didn't need it.
There is, yeah.
I don't know if this is on or not. And so I'm sort of curious why that notion isn't needed at all.
Yeah, that would be at way, 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 way lower stresses, which is something we never
so we're
reached.
well
So
past
there
that.
is no,
Okay,
yeah.
so
So these, so if you remember the rheology, I can, right? Whoever is from the Raman Institute. As a function of, stress, then you have that slope of 1, but then your yield stress will basically be down here, right?
So this is all happening far above.
Yeah.
Okay.
So we're always.
Okay.
Yeah, I think one thing to emphasize is here the solid is being created by a shear rate.
Yes.
It doesn't exist.
Exactly.
Without the shear rate.
Yes. So it's really flow induced.
You need the flow to make it a solid.
Exactly. Yes. Yes.
It's not easy.
Yes.
The shear rate.
Exactly.
Just one clarificatory question. So what are the length scales we are looking at? I forgot. I think you already told us.
So usually there's a scale bar here. So this is 2 centimeters. So our entire container is 10 by 10 by 15 centimeters. There are effects of the wall at some point. If these bubbles grow very large, I'm ignoring them in this talk.
Yeah, here, even if you increase the rate of air injection, I don't think you will observe system spanning crack because I think your suspension is not confined.
Absolutely. So the only thing
Right.
I can say is what's happening right at that interface.
Right.
Now, I have no idea. So up here, we know it's fluid-like. Sometimes you have these bubbles that someone was
Yeah.
asking about. They're actually really nice because they show you what can flow, right?
Yeah.
Yeah.
No, because I have seen a paper like in 2D Hileshaw.
Yeah.
So they are like at high rate of injection, you get fracture.
But it's different from here because we don't have that confinement.
That confinement.
Yeah,
If you
because
probably put a like solid confinement on the top or like rigid
absolutely
confinement.
nothing would happen because in the Healy-Shaw cell, what's nice, so Healy-Shaw cell is doing this experiment between two parallel plates, and then your stuff can escape to the outside, right? Here we can't quite do an equivalent of that because if we
I see.
put the
Then,
top,
okay.
then
The pressure
then
will build up
nothing.
simply,
Yeah.
Okay, thanks.
All right, so just in the interest of time, how are we doing actually? You guys are wonderful in asking questions. Oh, good. So if that's fine for you, I'm going to ask you to keep the rest of the questions, which I can tell you a second story. And the second story is also about flow-induced structuring of a complex fluid. But this time what we want to consider is an entirely different type of complex fluid, a liquid crystal. And so the type of liquid crystal we look at are disodium chromoglycate solutions. These are lyotropic chromonic liquid crystals, which means that they are water-based. They're composed of disc-like molecules that self-assemble into cylindrical ergets, where these cylindrical ergets then form the liquid crystalline units. In this talk, we're going to prepare a 13% DSCG solution. At this concentration and at room temperature, the material is in the nematic phase, so on average, all the ergets point in the same direction denoted by the director N. So the material has an orientational ordering, but no positional ordering. We place this DSCG solution in a microfluidic channel with a plate spacing of about 8 micrometer. We're going to apply a pressure-driven flow and see what the system does as it is pushed out of equilibrium. Now, I want you, for the rest of the talk, to remember two things. The first one is that we prepare the surface anchoring conditions such that at rest, the directors are planar, so parallel to the plates, and aligned in the flow direction. The second thing to remember is our choice of coordinate system, where the flow direction is in the x direction and the cell thickness direction is the c direction. So we're now applying if we now visualize the system at rest in this alignment on the crossed analyzer and polarizer, then it will simply appear black. Now we're going to apply a constant flow rate, push the material out of equilibrium, and what it does is something that to me is truly quite amazing. It forms these large-scale, fairly ordered stripe-like patterns. And what I want to tell you in the next 15 minutes is what these structures are and why they are a bit remarkable. And so in order to see what's happening here, I need to tell you about two quite peculiar properties of these lyotropic chromonic liquid crystals, these DSCG solutions. And the first one is that this is a so-called tumbling nematic, which means that there's a non-zero viscous torque acting on these ergets for any orientation of the director in the shear plane. So essentially this means, in simple words, that shear very easily destabilizes these nematic solutions in the shear plane. The second quite peculiar property is that these materials have a high elastic anisotropy, where of the three different deformation modes of a nematic liquid crystal, a splay deformation, a twist deformation, or a bend deformation, twist is by far the cheapest. So the twist Frank elastic constant is about a factor of 10 lower than the elastic constants of splay and bend. As a consequence of this exceptionally small twist Frank elastic constant, upon the onset of a flow, the material will try to avoid the costly splay or bend deformation that it would undergo when placed in the shear plane. It does so by reorienting by a twist deformation to adapt a so-called log rolling state where the alignment is pointing in the y direction. Now, this log rolling state is really the least compatible possible with our surface anchoring condition. And to resolve this incompatibility, what the system decides to do is to form these stripe-like patterns. And so to see what these stripes are, we're going to zoom in into a region of these stripes and use a couple of different visualization tricks to see what the director field is that makes up this structure. So the observation that these dark stripes remain dark upon a rotation of the crossed analyzer and polarizer by 45 degrees indicates that at these dark stripes, the director is in a homeotropic alignment, so pointing parallel to the cell thickness direction. Using a full wave plate optical compensator allows us to distinguish between aggregates aligned parallel or perpendicular to the flow direction. We see that they are between the dark stripes predominantly aligned perpendicular to the flow direction, which suggests a twist deformation along the flow direction. So we have a twist deformation along the flow direction. We also need to consider what's happening in between the two plates, where we can use fluorescence confocal polarizing microscopy to show that there's also a alternating twist deformation along the gap direction. But this prevalence of twist deformations is a consequence of that small twist Frank elastic constant. So putting all of this together and simplifying our notation, denoting a director pointing in the z direction as a line, in the y direction as a dot, we see this twist deformation along the flow direction, a alternating twist deformation along the gap direction, which gives us this, to me, a really elegant periodic double twist structure that spontaneously forms as we push the system out of equilibrium, and that gives this characteristic period of these stripes.
All the twist signs the same?
Yeah, we don't know. So experimentally, we can't resolve that. We collaborate with Ri Chang at the Hong Kong University of Science and Technology. In his simulations, he sees a 50-50 chance of picking a hand in this or a.
And I have no expertise on chronomics.
Mm-hmm.
I think I've heard that K24,
Yes.
which is a saddle splitter, plays a very important role in these systems.
Absolutely, and that's really what allows us to understand what's happening here. Yes, yes. No, so it's distinct from a blue phase. So it's really a double twist in the sense that you have twist in the flow direction and alternating twist in the gap direction. And it's a material that is, and I'm coming to that in a second, is achiral itself. So there is no pitch length intrinsic in the material. So it's really just flow-induced structuring. No, it's not the same. So you don't get this double twist in the blue phase. Yeah. Yeah. So these stripes, they happen over a range of flow rates of about a factor of 10. It corresponds to the range where the Ericsson number is of order 1. And which Ericsson number gets clearer in the rest of the talk. So if I push faster, then the system destabilizes and forms a specific type of defect, pure twist disconnection loops.
And what if you increase the speed beyond that? range field align like show only homeotropic alignment?
So right after this range, if we increase the shear rate, there's a range of defects, pure twist disconnection loops that form. If we go even higher, then the system starts to form a sort of shear aligned state. It's not perfectly shear aligned, but it's close. Yeah. Yeah. All right. Okay, cool. So there's two things that we think make this structure quite interesting. And the first one, which I think you alluded to a little bit, is that this is a chiral structure. And so just to remind you, a chiral object is an object whose mirror image cannot be superimposed with the original, whereas an achiral object is one whose mirror image can be superimposed. Now, our twist-like structure is chiral. I have here my twist-like structure. I have the mirror image. We see that one turns to the left, the other turns to the right. Now, no matter how I translate or how I rotate my mirror image, I cannot make them superimpose. One turns to the left, the other turns to the right. And you're absolutely allowed to left. These are a little silly, so don't think they were serious. So the fact that these structures are chiral is actually really non-trivial. Because usually, in order to form large-scale chiral structures, there's two common ways of how nature does so. The first one is if I have a chiral input that in turn generates a chiral output. So, for example, if I prepare my surface anchoring conditions in a specific way, I rotate my top plate by 45 degrees, I get a rotated, twisted chiral structure, but only because my input itself was chiral. The other common way of how to break mirror symmetry is in the absence of a chiral input, if my chiral structures are made of chiral building blocks. DNA is maybe the most famous example. It's chiral and composed of chiral building blocks, the nucleotides. Now, in our system, we have a chiral structure that's made of achiral building blocks in the absence of a chiral input. So indeed, our DSCG aggregate is achiral. And so these DSCG solutions have been shown to be able to break mirror symmetry at rest if they are confined to a very strong spatial confinement. For example, if they're confined to tectoids or confined to pillars. However, in this case, at rest, the system is still achiral, and only once it's pushed out of equilibrium, it forms this shear-driven chiral structure. And so I want to show you what the mechanism is that leads to this mirror symmetry breaking. The second somewhat peculiar characteristic is that these chiral structures possess a well-defined length scale, this period of the stripes, even though there's nothing in the system that imposes a length scale, and I want to show you what selects it here.
Not even the thickness.
The thickness is going to play a role, but is indirectly coming into the argument I'm going to give. Yeah, yeah. All right, so why do these form? So it's helpful to zoom out a little bit and look at a larger region, where we see that these stripes coexist with uniform regions. Now again, using a full wave plate optical compensator shows that these uniform regions can either be blue or yellow, which denotes distinct tilts of the director field in these two regions. Now, whenever I have two regions coming together by a divergence plate of a configuration, I see a sharp, stable line. This is a stable domain wall. On the other hand, if they come together as a convergence plate configuration, the boundary becomes unstable and develops into these stripes. Now, these two configurations cost the same amount of energy, yet one is stable, the other is unstable and forms the stripes. And to see why that is, we again have to consider what happens in between the two plates, where because of our pressure-driven flow, we have a Poiseuille-like velocity profile, which gives us a non-uniform shear rate across the gap, which induces a divergent splay deformation between the gap. Now I'm going to give you a bit of a headache. So if we now put together this divergent splay from the side view with that from the top view, we get this so-called double splay configuration. Here's our attempt at building a model. The sticks are the liquid crystals. The blue stuff is the surrounding. Where the important thing to note is that if I consider two orthogonal planes, I have in both orthogonal planes a divergent splay deformation. On the other hand, if I put together this divergent splay from the side view with the convergent one from the top view, I get this biaxial splay deformation. This is even harder to visualize. The important thing here is if I now consider two orthogonal planes, I have two opposite directions of splay deformations. Now I'm going to spare you the math and some of the details, but this is where K24 comes in. We're going to do a stability analysis on these directed deformations at the boundary of the domains. But we can show that indeed the directors in this double splay configuration are stable to small perturbances and will go back to that configuration, which leads to this stable domain wall. Whereas if I perturb the directors from this biaxial splay deformation, the system will become unstable to these perturbations. It will develop into the lowest accessible energy state, which we can show to indeed be this periodic double twist structure, which is the cheapest because of that smallness of the twist-free anchinelastic constant. In this stability analysis, K24 comes in and plays a big role. So this gives us a unique pathway to mirror symmetry breaking, where we see that flows can both induce and stabilize these chiral structures. So finally, to finish, I want to show you what selects a length scale in this system, where experimentally we see that this stride period decreases with a power law with an exponent of -0.5 with the average flow velocity, and exhibits systematic dependence on the plate spacing. Now, to understand why these dependencies occur, we have to consider the deformation modes in this structure, which are predominantly twist, but also in these red regions here, we have bend deformations. Now, given that bend is much more expensive than twist, we hypothesize that the selection of a length scale is given by the competition between the bend elastic torque from the director field and the viscous torque from the shear flow. So to probe that hypothesis, we write down the director field in the regions of these bend deformations. We can write down the Nemeada dynamic equation in this region, which tells us about the direct orientation in space and time, where I have two terms, the viscous term and the bend elastic term, which I can express by scaling analysis with parameters that we can measure in the experiment. Balancing the two terms then gives us a prediction for this critical period from the competition between the bend elastic torque and the viscous torque, which indeed shows a 1/√² dependence with the velocity and a dependence on the gap spacing, where I can now normalize my stride period with this critical stride period and get my data to collapse onto a master curve. So the selection of a length scale in these liquid crystals is due to the competition, or the period is selected by this competition between the bend elastic torque and the viscous torque. This leads to these characteristic period is the period of these double twist structures, which break mirror symmetry in an achiral liquid crystal because of these shear-induced biaxial splay configurations. So with this, I want to introduce the people who did all the work. So the liquid crystal work is work by graduate student Ching Chang in collaboration with Ri Chang from the Hong Kong University of Science and Technology and Chuang Zhou from UMass Amherst. The work on the fractures in cornstarch is done by Paul Leland in collaboration with Jean-L Corey from Schlumberger and Ivo Peters at the University of Southampton. And so in the interest of time, I'm going to skip a little bit of music. You can ask me about it. But I hope I could show you. You look. I know. Do we have a minute? No.
Do, yeah.
Yes? OK, then I'll show you. So one thing that has really been fun over the past few years is that I get regularly the chance to essentially collaborate with Multiverse, a concert series by director David Ebert, where we collaborate with different musicians, visual artists, other scientists, dancers, in order to present our work to a broader audience. And so we recently, right now, we're actually working on a performance based on the fracturing work that I showed you in the first part of this talk. The last show we did was on fluid instabilities in these liquid crystals, where you here see a 50-second example of what we do. So this is really the end of the talk. I showed you two examples of how flows can induce structuring in complex fluids, where we show a structural signature of the shear jam state that occurs upon the fracturing of the in dense suspensions. And I showed you a unique pathway to mirror symmetry breaking in pneumatic liquid crystals. I'm happy to take all your questions tonight over dinner and for the rest of the week. Thank you.
Yeah, one or two pressing questions. If not, we can.
Have you done any experiments in circular tubes?
With the liquid crystals?
Yeah.
No, but we want to do that because then you can actually get the...
Yeah.
Yes.
And the second question is, are you sure that whatever be the shear rate, your composite particles are right circular cylinders rather than something like the Pisa Tower, you know?
So
That may change the elastic ratios.
molecules themselves are kind of disc-like, and they stack on top of each other because of pi-pi interactions. Okay, and so
shear,
I think that
if your shear rate is very different in different parts of your...
Oh, yeah.
There is a possibility of a change of shape, and that may change the elastic
Yeah,
ratio.
so these materials are known to, if you go to even higher flow rates, these aggregates can break. And then you see a transition to an isotropic state. I don't know about the Eiffel Tower. Maybe we should chat more.
But I don't know if the shape can change.
Yeah, I'm not
Because
sure.
That can change the elastic
Yeah, I don't think people have reported it, but I don't know. I mean, we can't visualize it.
Yeah, so I missed something. What is the experimental evidence that you have chirality in the system?
Yeah, so we know experimentally that we have twist in both of these directions. If you put that together. Yeah, but then if I do have this periodic double twist structure, it is chiral. So
Yeah, but what is the experimental evidence for that?
our different optical visualization modes that show that there's twist.
If you send polarized light, for instance,
Yeah, we're doing that right now. So the other way of probing it is to say, can I now actually use these structures in order to detect chirality, right? So if I put in, yeah, we're working on all of that. Yeah. I know. What if I put bacteria that can actually swim in there? Will they swim that way, right? It's fun, yeah.
Okay, so let's thank Imran for a very nice talk. So our next speaker is Ambarish Ghosh from the Indian Institute of Science, and he'll tell us about magnetic nanoswimmers.
The boundary effects here, so all the data I stopped was before we see boundary effects.
Thank you.
They will influence the growth. Yeah.
Thanks for the introduction, Vijay. Thanks to the organizers for inviting me and allowing me to present this work. I realize...
One second.
Sorry.
If it's too cold, maybe you switch off the AC. Some people are putting on their caps. Or increase the temperature a bit higher. Yeah. Shriram ran away. Sorry, Amrish.
No problem. Here? All right. All right. Yeah, so I'm from IISc Bangalore, and this is the research we have been doing on these colloidal particles over the last quite a few years. So I should first acknowledge the large number of present and past students who have helped in this research program, and my collaborators, the ones highlighted and the ones whose results I'm going to show today, from diverse backgrounds ranging from material scientists, biologists, doctors, so on and so forth. So I'll spare you the mumbo jumbo, but the key point that we want to do is realize, at least this was the original motivation, to get these active nanoswimmers or robots or propellers, whatever you want to call them, and actually move them in vivo, in living systems, and to carry out a few tasks. Just to sort of tell you the crux of the research program, we want to move small things in fluids and gels, but we want to, of course, do it with good control, but we also want to do it from a distance. So I should clarify that we are not talking about optical tweezers or magnetic tweezers. So we are using forces or torques that do not have a spatial gradient. Okay, so we are talking of spatial uniform fields when we want to move these small objects. We want to give them multiple functions because this is what, well, he said, and later the folks in the movie carried out. We want to give them multiple functions, not just in isolation, but perhaps also in collection. And then it's important to realize that the applications are not necessarily biological, although you can do quite a few biological applications. And in fact, I'm going to tell you about some biomedical applications that have actually come out from the group and which we are trying to commercialize at this point. So I don't have to tell you about this picture. This is from Purcell's very famous lecture. Effectively, what we are trying to do, our strategy of active nanoswimmers, the artificial active nanoswimmer is based on mimicking the motion of a corkscrew. It's just a mimic. It's not really like an E. coli bacteria, but the key idea is we make some artificial screws, we give them a magnetic moment to it, that's the handle, and then when we have a rotating magnetic field, the screw rotates in sync with the field. So the screw rotates, and therefore it translates similar to the way, similar, not the same, a way an E. coli moves. So we do not have a counter-rotating head and stuff like that. We are talking about a rigid helix rotating and moving. I'll go right into the way we carry out these experiments. We use a technique called glancing angle deposition to make these structures. effectively, it's a surface at a very extreme angle with respect to some material that we evaporate. And because of the extreme angle, any material that comes and hits this substrate forms some form of a porous nanostructured film. And then by manipulating this substrate, you can make a wide variety of different shapes and structures. So this, for example, is the structure of a helical structure, where the scale is of the order of 5-6 microns long and about a micron wide or so. Typically, the experiments are done by creating some form of a rotating magnetic field in a triaxial Helmholtz coil. And then if you take these structures and put it in a little fluid and you make them rotate, well, they will move. Yeah.
Beautiful, but in a patient, you would have to put your cords much further away.
Absolutely, absolutely. So the point here is, and in fact, we already have it. Right now, we don't have human patients, but we have some mice. And the mice patients, still small, but it's actually scales as 1 over the dimension. So that's the and that sort of saves us, you see. So it's, exactly, right. Absolutely. Absolutely. So you do not need to have an NMR size structure. And also, if you're talking about things that are only concentrated on a head, then that's something we can do in our lab right away. Okay. Yeah. So that's the way this thing works. So just to tell you how the swimmers move, well, effectively, if you just have a rotating field, the screws will rotate and translate. And just by controlling the magnetic field, you can make them go from east to west, north to south, and so on and so forth. And, you know, if you want to get some publicity, you can try to, well, this is similar to my last speaker, but this is just done in a microfluidic device. It's not real music. But then, most importantly, you're not limited to just one, but billions of such swimmers altogether. So what you are really talking about here are approximately one swimmer every 3 micrometer cube volume. So that's really a very, very large density of swimmers. And a lot of experimental efforts go into it to make it stable, to make the magnetic moment large enough. And I'm not really telling you all these details in this talk. So as time comes, I'm going to probably give you some of these experimental details. So just to give you an example, even something as simple as this corkscrew motion, even that should not be too obvious to you. So, of course, this is the animation that I showed you, in which the red is the magnetic moment, which is rotating in sync with the blue rotating magnetic field. But that's not the only way this object can move. So if you look at this animation, well, even here, the object is rotating in sync with the field. So rather than a corkscrew motion here, well, it seems that a tumbling motion is also equally probable. So which one does it choose? And we have worked out all of these details, and in fact, you do see this type of motion under the right conditions, and you can go this sort of corkscrew type motion, tumbling motion, some form of a wobbling geometry, and all of these things, we have sort of looked at it over the years as a function of field frequency, viscosity, elasticity, effect of the surface, magnetization, so on and so forth. So these actually lead to some very interesting applications, and so this is what I'm going to get to over the next couple of slides. The point of showing this is to just say that there are a lot of different things that can affect the dynamics, so one should not just assume the corkscrew to be the only way these things can move. So what are the key advantages of this experimental system? Of course, this helix structure, because of this way we are making these structures, you can have a wide variety of length and width. And this is very important if you're talking about moving in biological media, because something out there, because it's a very porous material, the thickness of the helix plays a huge, a very important role. So out here, this is one of the largest structures that we have made, and this is one of the shortest ones. Just to tell you, the thickness out here is about 700 nanometers, while the thickness out here is 50 nanometers. So you have this wide range of geometries that are accessible to you from an experimental perspective. So thickness of the helix is a very important aspect. You can have wide different types of materials and configurations. So you can put your magnetic material on side, or you can put the magnetic material while you grow the helix. You can put your structures, make your structures, and then coat it with something all on the surrounding. So you can play all these different tricks, and depending on the type of things that you would like to carry out. And very recently, we are very interested in using diamonds, little nano diamonds, to attach them on the helices. And I'm not going to show you any experimental results, but the idea is, can this diamond be used to sense the local environment? So what I'm going to tell you next is give some examples of local measurement of physical properties using these colloidal particles, and also tell you a little bit about how these things can be used to, for some form of delivery or therapeutics in highly constrained environments. So that really is the plan of the talk. I'm going to first tell you about how we use this as local probes. So perhaps the most easiest way you can imagine this to be used as a local probe is simply look at its velocity as a function of the field frequency. And as you would imagine, the velocity should just go up linearly as a function of as a function of frequency, assuming this is a Newtonian medium. Beyond a certain frequency, the rotating object cannot keep up in sync with the torque, so as a result, there'll be a step out and it goes down. And the frequency at which this step out happens will be inversely proportional to the viscosity. So this will be the simplest way in which you can measure local viscosity. The key point is your sensor is maneuverable, so you can move from point A to point B and actually measure local viscosities. Now, of course, this will not work if the medium is non-Newtonian. Just to give you an example, this is an old movie we took when we put the swimmers in blood. And as you can see, the nice movie and the G-clef sign, all of that, well, it doesn't happen in blood because you are really in this highly constrained, well, almost jammed media. So it shows some form of a stick and slip motion when you are in a colloidal suspension. So how you deal with the motion of these objects in this type of non-Newtonian medium is an active area of research, not just by us, but many people. And so, for example, there are theories and experiments which suggest that this linearity of viscosity, of velocity and frequency breaks down when you are in a non-Newtonian fluid. So the pitch, the effective hydrodynamic pitch of the helix can be a function of so here the local time scale, which is given by the frequency of the medium. And, but the importance of using this as local rheometers can be seen when you use some device where you go from point A to point B. So just to go a little bit into this detail of how you can use this as local rheometers, I've already told you about the various type of dynamics it can happen. So it can have a tumbling dynamics at some frequency, and then depending on the frequency at which you make it rotate, it can go into some form of a corkscrew dynamics. And these equations you could derive simply by solving Newton's laws, and you can see how the theta, which is the angle at which the whole thing wobbles, changes as a function of frequency. The important aspect is this wobbling angle is a function of the applied torque, the magnetic moment times the field times some geometrical parameter which gives which direction the moment is with respect to the helix, divided by eta is the viscosity, omega b is the applied frequency, and gamma short is a geometrical parameter which tells you the frictional coefficient about the short axis. Okay, I think about the long axis, I think this is incorrect. But the key point is the sine theta goes as 1 over the viscosity. And this already tells you that if you simply make these objects move and just observe their dynamics, then on the fly you will be able to measure the local viscosity. So this is the experiment that we did where we took it from one fluid to another, and these were miscible fluids, just in case you were wondering. There is no hard barrier between them. But then what you can do from here is to actually make a viscosity map at a time scale much faster in which the fluids mix together. So this is an active measurement probe that you can use to measure, for example, local viscosities, and I'll tell you later, in principle, you can also go to elasticity measurements, and that's work in progress. You're not entirely limited by Newtonian fluids. Out here, the same technique can be used for shear thinning media as well, and using exactly the same math. Now the question is, well, now that you have this tool, what would be a good place where you can make these measurements? What is the example of a very nice, complex, and heterogeneous environment? And when we thought about it, it seemed to us that the best example of a heterogeneous environment that's also rheologically quite complex is a living cell. So the experiment that we did was to take these swimmers and incubate them with living cells. And it was not too surprising, but the cells sort of internalized them. The surprising part was the swimmers continued to be mobile inside the cells. So what I'm going to show you here, this is a confocal image of the swimmers incubated for 24 hours inside cell. And this is basically, I'm going to take a stack of images, and that's the movie that you're going to see here. And you have to look at it a couple of times and look for these little red dots. So those little red dots correspond to one swimmer inside the cell. So the particular images that you're seeing here, there are two swimmers, and you will see two red dots, one here, one there. You see that? Okay. So the red corresponds to the swimmer, the blue is the nucleus, and the actin filaments have been stained here. Now, what's really interesting is that they get internalized is not very surprising, but what is really, okay, so that's just a 3D reconstructed view. What was kind of heartening to see was, well, the swimmers continued to be mobile inside the living cells, and in case you are wondering that the cell is dead, well, this is a movie that we managed to capture once, in which just when we were taking the movie, the cell decided to divide, but we were not lucky later. But now we have a system where we want to specifically make this type of measurements where one can look at the motion of the swimmers inside cells while the cell is doing something interesting, be it dividing it, or maybe it's some form of migration, and that's the type of experiments that we are now well catered to do. Just to tell you a little bit more about how these things move inside living cells, well, we are in Bangalore, so we know how to move in constrained medium. So the way you do this, you go forward, then you come back, and then you change your direction, so on and so forth. And that's exactly the way you also have to make these things move. But then again, you know, unfortunately, to raise funding, you need to make a little bit of publicity. Well, we decided to write the letter N inside the cell. And the way this was done was actually going forward, finding out an obstacle, coming back, changing the course, and all that. And this also tells us that just by looking at the way, you know, looking at the motion as a function of direction in which you make things move, you can actually make an obstacle map within the cytoplasm. A very, very important aspect to note here is that this helices has to be thin enough. If it's anything more than 200 or 250 nanometers, they will simply not be able to move inside the cell. And this just points to the approximate porosity of the cellular cytoskeleton that people have also reported in the past. Something that's very interesting that we have found out from our experiments is the hydrodynamic pitch turns out to few. So what you are seeing here is the motion of the swimmer inside the cell, and as it goes from here to here, you will see that the overall hydrodynamic pitch goes by approximately a factor of 5, while the Newtonian pitch out here is approximately 300 nanometers or so. So this factor of 2 increase in the hydrodynamic pitch points to the highly enhanced local viscoelasticity, local elasticity in certain parts of the cell. And from our measurements from many, many different cell lines, what we find out is this always happens when the swimmer is somewhere close to the nucleus. Of course, there could be other physical forces, there could be flow in the cell as well, but we have done back and forth, and this particular observation seems to be quite reproducible. So right now, the experiment that we are trying to do here is actually to make a rheological map of the cell. Of course, what we realize is we will never be able to make a real rheological map, because maybe it will be a combination of rheology and constrained environment together. But as far as I know, this has not been done. Of course, compared to passive microrheology, here you can actually take the probe from one point to another, which makes it somewhat powerful. Now, if you want to, while we were doing this experiment, we realized that, well, inside the cell, of course, has all these difficulties, but many of these difficulties go away when you are going outside and looking at extracellular matrix. So the particular experiment that I'll show you here, this is some form of a 3D microenvironment or some form of a tumor model in which we have seeded cancer and normal cells in Matrigel. So Matrigel it's like a gel, and basically the normal and the cancer cells have been grown here and incubated, and the swimmers are simply put inside this Matrigel-like environment. Here we saw something extremely surprising. All that we did was to take the swimmers and make it go back and forth in this sort of a 3D microenvironment, and after doing it for about an hour or so, we found out that the swimmers were all somewhere stuck near the cancer cells, while the swimmers which were near the normal cells were happily moving around. When I say near, I do not mean right at the surface, but even about 10-20 microns away, they were all stuck near the cancer cells. There seems to be some form of a adhesive force between the environment surrounding the cancer cells along with to the swimmers, which was not present with the normal cells. So we tried to, it seems that the cancer cell surroundings are sticky, and this is not completely surprising, because we know that the cancer cells remodel their local environment in some ways. So it seems that what we are looking at is, in a way, some form of a local remodeling by cancer cells that is picked up by the motion of the swimmers. And I'll tell you a little bit more about what we have measured here.
These were cancer cell lines on a slide, so, or tumor explants.
So this is effectively a very thick gel, matrigel, more than few hundred microns, okay, in which cancer and normal cells were seeded.
Okay, thank you.
So one of the things we could do, because we know the magnetic moment very well, we could see, well, once they are stuck, what is to get them de-stuck? So in other words, we could actually measure the adhesive force between the swimmer and the cell, and what is shown here is the force of adhesion as a function of distance from the cancer cell surface. So what you're seeing here are different types of cancer cells, MDA, MB231, MCF70, 42, HC70, and of course with the normal cells there was no adhesive force whatsoever, so that's just zero. So if you look at the force as a function of distance, you can perhaps say that, okay, it has some form of an exponential dependence. The overall length scale over which this remodeling happens turns out to be anywhere about 20 microns or so. What is kind of interesting is that characteristics force does depend very strongly on the cell line, and we don't have a good idea why, but it seems more aggressive cancer cell lines have a much stronger adhesive force, characteristics force that's associated with this behavior. To try to understand what causes this adhesion, the experiment that we did was to modify the surface of the swimmers with PFO, which makes it extremely strongly negatively charged. And in that particular case, as you can see from this experiment, the ones without the PFO coating get stuck somewhere near the cancer cell, while the one with PFO happily roams around. So it seems that the origin of this adhesion is electrostatic in nature. At least that's really our best guess. Now, what is the cause of having enhanced electrostatic interaction of the cancer cell surrounding? Why are the cancer cell surroundings differently charged compared to the normal cells? And here our biologist colleagues created this assay in which they suspected that it must be the highly the hypersialylated proteins around the cancer cells, or in other words, the sialic acids that's present in the environment around the cancer cells that causes the charge environment to be very, very different. So what you are seeing here, this is effectively a fluorescent labeled sialic acid distribution in MCF7, and it is known that the sialic acid distribution in these cell lines have an anisotropic behavior in which spatially the sialic acid only goes in one side of the cancer cell, and when we do our mechanical motion, when we took the swimmers and see if they are getting attached or not, we see something very, very similar. So the cancer cells on this side do not get stuck, while the swimmers on this side do not get stuck, while the swimmers on this side do get stuck, just like the way basically it shows the same type of anisotropy that's shown in both the sialic acid distribution as well as the adhesive force distribution. In a different cancer cells, while the sialic acid distribution is spatially uniform, we also observe the swimmers to be bound on both sides of the cancer cells. So you see the same type of isotropy in the adhesion force. So effectively,
Ambrish, is the statement then that the sialic acid distribution determines the ECM distribution?
So we believe, the sialic acid, what we have imaged here is the spatial distribution of the sialic acid in the ECM. And we believe that the charge-based adhesion that's happening is between the less negatively charged helical swimmers and the sialic acid. So this is the part where things are getting stuck, and also the spatial distribution is not too far from what we have seen in our mechanical assay. That's the basic idea. Okay. So we believe that this is kind of new and interesting because in principle this can lead to a new type of a targeting strategy in which you actually don't need to see where your colloidal probes are going, but still be able to sort of cluster around the cancer cells. So this is an experiment that we are trying to do in animals right now, and to try to see if even in a sort of tumors in mice, whether some similar sort of behavior is observable or not. So I'll show you a little bit along this direction right at the end of this talk. This is just one slide. I wanted to tell you about something non-biological. So you have all heard about plasmonics and plasmonic tweezers. The key point is if you have sort of gold nanoparticles, and if you, let's say you put this whole thing in a fluid and you shine light, well, because of plasmonic effects, there is a strong local electric field and a strongly enhanced gradient field that gets concentrated around those gold pillars. So if you have some particles in the fluid, they might get attracted and get trapped here. So this is the basic idea of a plasmonic tweezer. And the most interesting aspect about this technology is the tweezing happens at much, much smaller optical powers compared to standard optical tweezers. And, but then, of course, a big problem with plasmonic tweezers is that your local field is only enhanced in this tiny region. How would the colloid know that it has to go here? And I guess you can imagine what we did here. All that we did was to couple this with our magnetic swimmers, and effectively we took these helical swimmers, put some plasmonic particles in there, and then when you shine light, they work as kind of nice little movable plasmonic tweezers. So here we turn on the light, they will effectively be able to trap the particle, and then subsequently we can take it to a region, and then if you reduce the light intensity, they're going to release the particles. So it's simply a very simple demonstration of a trap, transport, and release type of a mechanism. But the good thing about this is this can actually work with a very large number of particles and swimmers all together. So I just wanted to mention this because many of these things are being now actively used for biomanipulation. A lot of people are trying to do this with this type of driven probes. Something before I change my topic a little bit. So right now the key aspect that we are trying to do here is we have integrated the swimmers with diamonds, and one of the key things that we want to do is to make a mechanical mapping that I've told you already, but also these diamond sensors. They are wonderful quantum fluorescent probes to give information about local temperature as well as local magnetic field, so they can be used for local NMR, and this is something that we are trying to do inside biological environments. Hopefully next time we'll get a chance to actually show you some new results of actually being able to do NMR inside a living cell. I want to, yes. Sorry?
What is the power of light source used for plasmonic tweezers?
So the effective intensity that we use is extremely small. So you're talking about less than a milliwatt per micron square. So if you do the numbers.
So how do you move it precisely around the cell?
Sorry?
That light source?
So the light source we are not moving. It's a global illumination. We can just turn it on and off to ensure that the local electric field around the plasmonic particles get enhanced.
Okay, so the movement of that trapped particle is not precisely controlled then, right? Because it's a global field.
No. So the optical field is global. The motion of the swimmer is perfectly controlled using magnetic fields.
So still it's rotating within
Absolutely. It is rotating and able to pick up and transport it. That's the basic idea. Okay. All right, I want to go a little bit onto this active versus driven part, and I want to tell you something boring about our swimmers. And effectively, these things are all externally powered, and because you are using a rotating magnetic field, all of these objects are, the direction of motion is completely governed experimentally. It's a zero force but a non-zero torque system, so it's a great system when you want good control. So you have all of them move all together in a certain direction. As opposed to active swimmers, where, well, the source of energy is internal, the direction of motion is either governed by interactions or governed by orientational diffusion, and it's a zero force, zero torque system. Perfect randomness, but of course out of this randomness, a lot of interesting things emerge. So the question that we wanted to see is, can we combine both, and can we basically turn it on and off whenever we want? So can we have both control and randomness? And the idea there would be, can we use as fuel small magnetic fields, but the direction we do not control externally, but let it come just out of interactions with each other and surrounding. That will be a way of making an artificial active matter, and of course the hope is to see this sort of things happen out of this system. So how do we make this system active? And the answer to this was, well, if you want to decouple the fuel and the direction, you can start with this swimmer. This is the swimmer, and now this view, you are looking at the swimmer at this end. If you apply a magnetic field in the positive direction, the magnetic moment is pointed upward. Subsequently, if you reverse the magnetic field, make it pointed downwards, the magnetic moment will be pointed downward, and then if you just give it some form of an oscillating field, well, it will make a turn, go down, and the turn can happen either through clockwise or through counterclockwise manner, and in the presence of an oscillating field, it's again going to go back up. So the main idea is it will go like this, and then again it will turn. Every time it turns, it will have a small linear motion because it's a corkscrew. The key point is, this is, in a way, giving it a small motility, but at no point of time you are telling it which way it can point. And that's the key idea. You have decoupled the direction of the motion with respect to, so basically if there are two of them, they will happily go in this sort of back and forth motion, pointing in their own different directions. And that's the key point, that there is no restriction in the XY plane, while it can effectively slide in the XY plane in any pointing in any one direction. And that's really what you see experimentally. This movie for some reason is not playing very well, but effectively is doing some form of a back and forth motion. In other words, it only goes in one direction and it comes back in the other way. Just to tell you how this happens, that's because of a slight weight asymmetry that's present in the system. When you have a magnetic field pointed upward, the screw points in this direction. When you change the direction of the magnetic field, it gives a clockwise rotation, and as a result, now the moment is pointed downward, and then when you turn the magnetic field back on, it again goes back. So, bottom line, it just does something like this. It goes forward, goes backward, okay? And this happens due to the combined action of magnetic field and some form of a weight. So the good news is, indeed, it has powered by magnetic fields. Its direction is not specified, but the bad news is, well, at the end, it's still doing some form of a back and forth motion. So in Purcell's language, this is just a reciprocal swimmer. That's it. Just to tell you, well, reciprocal swimmers are not completely bad. They also show a little bit of enhanced Brownian motion. And the reason why this happens is because every time it does back and forth, it also has some diffusion in the XY plane, some rotational diffusion. So within this back and forth motion, it does manage to actually have a slightly longer reach compared to a passive object. And this is really because of this rotational diffusion in the XY plane. But still, how would you break the reciprocal symmetry? And we again thought about it for some time, and the answer was actually very, very simple. I already told you about how this happens. Basically, the field is pointed upwards. When you turn it back, due to the weight, it sort of slowly goes and gives a clockwise turn, and now the moment is pointed downward. Well, the idea would be to actually make it a very fast switch. If you make it a very fast switch, then effectively both of these are equally probable. You can either go back by a clockwise or you can go back by a counterclockwise rotation. So if you keep on doing it, on the whole, there will be slightly more clockwise turns compared to the counterclockwise turns. And I will actually spare you the details of this. I do have the slides, but effectively this is nothing but a Brownian ratchet that we used to effectively break this reciprocal symmetry. So just by controlling the time scales in which you vary these magnetic fields, you can actually make the swimmer go from reciprocal to non-reciprocal. Okay? So I will actually spare you because I think it's a little bit the end of the day. So I'll not go through all of these, but the key point here is, well, so that's just the way how the ratchet works. And of course, for thermal ratchets to be present, you need to have a little bit of noise in the system. The most important method out here is that indeed when we did the experiments, we did find out that, you know, when you have this type of an asymmetric actuation in the system, depending on the ratio of the timescales, you can actually go all the way from a passive to a reciprocal swimmer to something that is completely non-reciprocal. So you can actually have an active Brownian particle based on entirely magnetic drive. And you can actually fit this to this mean square displacement, to time, and actually get some form of a self-propulsion speed, and you can get Peclet numbers which are slightly more than 1 within our experiments, and we are trying to make it slightly more. Now, it'll be nice to, and now for the next 1 or 2 slides, I'm going to tell you a few things that we don't have any idea about. Now that we have some active swimmers, it's also important to ask, well, what will happen if you put a large number of them? Well, they're going to interact. At very close distances, of course, the magnetic interactions will take place. But for these swimmers, far more important is the flow. Now, remember, we are talking about effectively a 2D system. And so the flow of this helical swimmer in bulk gets strongly modified when you are very close to the surface. And we have looked at this in collaboration with Alex Lesansky in some time. And to a very, very good degree, you can approximate the swimmers to be long cylinders. And you can develop a theory based on a distributed system of rotlets along the length of the cylinder and actually come up with an analytical formulation that matches very well with both computation and experiments. And effectively what you see is a rotlet, and then there will be an image of a rotlet and a few more higher order terms that all comes from Blake's theory. So the point of telling this is indeed these active swimmers are also going to have strong hydrodynamic interactions, and although we don't yet have a very formal theory of this, but I think there are interesting things one can look at in suspension. So I'm going to show you some movies here, and this is just a collection of propellers. What I mean here is it's a rotating field, so there's a large number of them, all of them sent in the same direction. And now, if you play this movie, well, what you're seeing here is a large drift in this direction. Well, this is not the direction in which we wanted it to go. We wanted it to go in this direction, but then because of the large fluid flow that it generates, it's all pushed to one side. Okay, and that's again not very surprising, and it matches with what we actually made a little bit of a little toy model, and basically what we get is a large drift, sideways drift, which depends on the local density. And we believe this density dependence can actually be used if you want to get a large collection of these swimmers and maybe focus into one point and things like that. So it's a nice little handle. Rather than this sort of a rotating field, you can use your oscillating field and get this collection of swimmers and just put some tracer particles, and as one would expect, indeed, the overall diffusivity of these tracer particles goes up, which depends on the activity and the density. That's not too surprising either. The part that we don't have much idea about is what happens when you have a collection of non-reciprocal swimmers. Now, these movies sometimes give a problem. So you can probably see a little bit of a motion, not anything, well, if you look at it from a distance, maybe try to look at this guy somewhere out here. What you will see, if you stare at it a little bit closely, is there are certain swimmers which continue to circle around in the same place over a long, long period of time. And this sort of observations we don't have a very, very good explanation of yet. We tried to make a similar model that we did, and at least we believe that something similar should happen, but a far more serious theory is needed to understand this business. if you have a large number of such collective swimmers and put it in a big cell, this movie is really, really difficult to see, but maybe if you concentrate in this region, you will see that suddenly a large number of dots all come together, and then again they disperse all very suddenly. There is no chemical fuel. All that we did was to apply the magnetic field and just observe it from a distance. And you can do these experiments for days and weeks. You don't have to do anything. So that's the great thing about this experimental platform. So hopefully we will be in a position to have a little bit better understanding from this. I must tell you this is work in progress. Okay? Hopefully we'll have more results to show in coming times. I have another 5-10 minutes, so I'm going to tell you a little bit about some applications that have come out of this business. So I've already told you about how you can make these things active, in which they have some effective self-propulsive propulsion speed. Now, one of them, of course, is their action in collection, in which they occupy and they cover a lot of area in a very low amount of, in very little time. And this does not even need any interaction between the objects. Just to cover a large area, large diffusivity can be extremely helpful. And one of the places where it's extremely useful, this is a picture of your tooth. So I don't know how many of you had root canals, but a big problem that happens in root canals is, well, you have to do it again. And the reason why you have to do it again is during root canal, and this is a picture I have, what the dentist does, the dentist drills inside your tooth, extracts all the pulp away, and then puts a little bit of bleach in your mouth, because there is a lot of bacteria that are present in this part, the white part, which are known as which is the dentin tissue, which is made of large number of small tubes. So we are talking about 1 million tubes. So you can imagine that this is the pulp, you have drilled into it, and out here, radially outward, all these little tubes are coming. And these little tubes are the place where the bacteria live, and these are extremely resilient bacteria. Even after the dentist has completely plugged it, they will be staying there and coming back after 2 months, and you'll have to visit your dentist again. The biggest problem with using bleach is in the short amount of time in these little tubes that we have, just about a micron or so and millimeters long, your bleach will not be able to reach to the depth of the tubule, and that's just diffusion. You are limited by diffusion of your bleach inside your teeth. So this is the part where you can imagine the swimmers are very good. The swimmers would be able to actively go in all directions and actually reach the bacteria. So, you're talking about 2 millimeters long, 2 micron diameter. So we need something that's small. We need something that's autonomous, meaning it will moving in all directions simultaneously. And you also want to give it some functions. So what you are seeing here, this is actually our experiments with human teeth. This is the pulp. This is a large number of confocal stacks of the tooth. This is basically the dentin tissue area. These are all basically dentinal tubules. So each of these images correspond to one such swimmer imaged inside a dentinal tubule. So effectively what you have done, you have put the swimmers here, turned on your magnetic field, and the swimmers all went in different directions. Now, we actually took it a little bit beyond just taking it all around. We decided, well, and the picture here, the green corresponds to the swimmers. Okay, so the two pictures have different color conventions. You have to be a little bit careful. So what you can do is you can take them, you can put them in by using this oscillating field, and that means it's a self-propelled system. But then you can use a rotating field and actually start retrieving them one by one. So this is the point where having both the control and the randomness is important. The randomness allows you to cover a lot of area in a short amount of time, and the control allows you to target or not target certain parts. The image that you are seeing here the green corresponds to live bacteria, and the red corresponds to dead bacteria. So what we did here, we again put the swimmers, and we only took it to this region, and then we applied a strongly varying magnetic field so that the region around the swimmers get heated up, and it was hot enough to kill the bacteria locally. So basically, we are talking about a new type of a therapeutic intervention, and this is what the startup started out to do. As we moved forward, we realized that there is an even bigger problem that you can address than root canal. So I don't know what your plans in the evening are, but many of you, if you go and have that cold beer, it's possible that there will be sharp shooting pain on your teeth. It's known as hypersensitivity. I don't know how many of you have it, but this is an extremely common problem when you have something cold. And the reason why hypersensitivity happens is if this is the tooth, this is the pulp, the inside, and this is the outside, there are actually little holes that are created on the surface of the tooth. So effectively what happens, the anything cold that you have is going to go and actually trigger the nerve ends that are present in the pulp. So this is typically what happens in slightly, well, I think once typically around 40 or greater age group, this starts happening. And basically the origin of this is that the enamel gets thinner. The standard solution to this problem is to use this toothpaste called Sensodyne, but I don't know how many of you used it. It stops working. If you stop using it, it stops working after about a week or so. So you're basically sold to them. And all that Sensodyne does is to create a small plug at the surface of the enamel, which goes away after a week or so. So what we did, rather than root canal, where we go in from inside and going up here and killing the bacteria, this time we took the swimmers and went from outside to inside. We went inside, and then I will spare you the details, but the key point was we made it actually go and block the dentinal tubules by having a little bit of calcium cement. So the swimmers from outside went in. Subsequently, there was some calcification that was triggered so that all these dentinal tubules got blocked. And we did it, that was the picture in human tooth. This was actually experiments. We have been doing things with animals, and this is kind of a funny experiment. I'll just tell you the result of this experiment. So we started with the mouse, and we gave both cold water and warm water. And of course, normally, baseline, a mouse doesn't really care. It's equally happy with cold and warm water. Subsequently, we brutally filed off part of the tooth to expose the enamel, and then it started disliking the cold water, started liking only the warm water. And then if we do not do any treatment, well, it continued to like the warm water and dislike the cold water. But when our treatment was done, they started liking cold water again. So so at least at a mouse level, we are in a good place. Just my last slide, I told you about this fantastic Voyager. Where are we right now? So we are doing things in animals, and this is a very recent movie that was taken in our lab. We took the swimmers, we put near-infrared probes onto these swimmers, and subsequently put them in the peritoneal cavity of a mouse, which is basically a fluid-filled sac, and it's actually connected to most of the major organs in the body. The easy thing about the peritoneal cavity is basically filled with a fluid that is effectively like water. It's not like a tissue, not like a gel. So it's very easy to move them, but the good thing about this was, I don't know if this movie will play. Well, yeah, so the movie is playing, and of course you can't make this nice single swimmer videos because you're actually taking an image in a living mouse while the whole thing is inside a microscope and a coil. But we did manage to controllably move it from one point to another at a reasonably high speed in a simple microscope. So this is still one of our small coils. So this is actually the evidence of, in some sense, a fantastic voyage. I think the point where this will become really useful, because our swimmers are extremely tiny, is if we can target real organs. And this is some recent experiments that we did in collaboration with our neuroscience department. And this is craniotomy, in which we injected the swimmers in the frontal lobe of the brain of a mouse. And again, we did manage to see some evidence of a motion. The reason why these things are moving, again, is because they are extremely tiny, so they are actually going through this gel. So the experimental system is extremely important here. So just to conclude my talk, well, what we have is a reasonably diverse colloidal system where many different fabrication schemes. It's very, very important to make it small. The smallness is important not only because you can move it in constrained media like cells and tissues, but also you can make it active, and that's very important. So the effect of thermal noise is very important. We believe it's a good probe for biophysics studies. I must tell you, we recently concluded some very important toxicity studies, because we hear this very often when we go and talk to doctors, is are they toxic? So the majority, the most of the material that makes these swimmers is made of silicon dioxide, and the amount of silica that we foresee that a patient will take is 1/10 of the amount of silica present in a pint of beer. So you're absolutely safe to have it. So with that, so these were published. These things are quite safe. And with that, I would like to thank you for your attention.
Yeah. Can you give some idea regarding the, I think part of the answer you have given regarding the how to apply the magnetic field inside the bio mouse you can somehow show, but just like in dental canal filling, do you have any idea what are the different ways to apply the magnetic field at that level in the living?
So yeah, I should have had, I don't think I have a picture here, but we actually have a tiny coil that fits in your mouth. So you just put it in the mouth and keep it for 10 minutes. It should work. So that's what the company has developed already.
No, this is, you need not take this question very seriously, but since you leave this particle, your swimmers inside your tooth, so whenever I have some problem, I just go and sit in a magnet for a while or what?
Sure. So in Feynman's talk, he does talk about implantable surgeons, so you can keep them permanently. So.
Hey, this is very fun to hear about. So I think I might have missed at the beginning of the talk why it's silicon dioxide. That's what grows nice chiral shapes. Would it be possible to expand this to using biopolymers that then just dissolve and get taken away, and you don't have to remove them?
Absolutely. Not easy. Not easy. The technique is better for inorganic materials, typically things that need electron beam evaporation to grow. But there are some recent experiments that shows that you can use metal oxide frameworks for this. We are yet to try it, but it's possible.
Not a physics question, startup question. Obviously, your primary target are teeth, and it's a nice target, but naturally, there's always the magic word cancer. So why? So bigger market, easier to achieve. So why startup in teeth and not in cancer?
Yeah, so that's actually a very, very good question. The cancer market, the regulatory approvals take at least 10 years, and the amount of money it takes to even get the first round of regulatory approvals is beyond what academics can do. You got to team up. Absolutely. So with the teeth, it's much easier. It's much, much easier. So we are already past the first level. So we should be able to try human trials sometime by next year. With cancer, it would have been at least an order of magnitude more and probably 10 years more. So that was the thought. Thanks.
No questions? I had one. So, I mean, you were telling us about the nanodiamonds that you can put in there, and they can sense the local chemistry.
Absolutely.
If it were possible to sense the chemistry of the local environment and use that feedback to change the movement, this would be like sensing other things or chemistry to bias your movement.
Sure, sure. So absolutely. I mean, depends on the time scale. Typically, the chemical sensing would probably happen with an averaging time of about a second or so. And then that's definitely within the sort of speed at which this control loop can work.
Sure. I'm thinking more like information processing mechanics. More questions, comments? Okay, if not, let's thank Ambrish for a very nice talk. And I think we conclude for the day, and dinner is at, Shantanu has some announcements.
Let's thank Vijay for chairing the session, and dinner will be at the same place as lunch. So, sorry? I don't know. They promised. Yeah, they promised that they will keep it ready by 6:30 because many of you must be very tired. So some of you may want to eat early and go to sleep. So 6:30 is what they promised. Yeah. I think it will go on.